Hello, welcome everyone. Thank you for joining this webinar today. I will give just a minute to allow everyone to join this virtual room before starting. In the meantime, thank you very much for being with us today. I'm Laura Viviani. I'm a consultant at SciEthic, supporting Humane Society International and the Animal Free Safety Assessment collaboration in the projects related to the biologicals products and the transition to non-animal testing for those type of products. So the webinar is going to start soon. I just really want to give a minute to the majority of the people to join it. We are having a lot of presentation today, so we will try to be on time. Make sure that you interact as much as possible with us if you have questions or comments. And there are a couple of rules for the webinars today. So you will be all in the listening mode as all the webinars. And if you want to have questions, please use the Q&A box that you find at the bottom of your Zoom window. I will monitor it and try to make sure to take as much as question as possible within the time frame of the individual presentations. I'm sorry in case we are not able to answer to all the questions, but the speakers, in case they are not able to reply live, they will try to reply you through the Q&A box or through the chat. This webinar is recorded. The presentations and the recordings will be available on the AFSA event page in about a week time. So please stay tuned. And if you have any questions, of course, about this webinar or in general, feel free to get in touch with me by email. So why we are here today? So the objective of this webinar is really to provide you the technical aspects, explaining how the transition to non-animal based vaccine batch release testing is possible. And how this type of alternative methodology has been successfully incorporated in regulation and guidelines for the veterinary products. We would like also to give you a lot of insights and references for the regulatory agencies, but also to the public and private laboratories and the manufacturers worldwide about how this transition is possible. At the same time, we would like also to show you what is in development, because of course the field is still working extensively to make sure that novel technology are going to be part of the future release strategy of this type of products. And of course, our idea is also to share with you how all these successful case studies or success to be case studies have a strong scientific, technical and data-driven rationale, and how that was successful in facilitating the regulatory acceptance of this non-animal testing. So why this workshop? So Humane Society International, that is an animal protection NGO, has been working since 2019 to promote the implementation of non-animal based testing for biologicals for both human and the veterinary side. You can see some of the activities through this website. And of course, a piece of the strategy of this organization through the Animal Free Safety Assessment collaboration is really to organize the multi-stakeholders workshop and also educational webinars like the one that we are having today. The agenda, it has been built in collaboration with all the experts from HealthforAnimals and their triage task force, but it's also based on all the type of discussions that we are having within the different industry and the regulatory authorities to really understand exactly what are the needs in terms of a better understanding on those technology and how the transformation from a regulatory perspective can happen. So before actually giving the floor to our first speaker, I really wanted to thank you all for being here. Thank the HealthforAnimals Task Force and all the speakers that are here today and sharing this interesting experience with you. And you can see here there are some upcoming events on the human side, but I would like to point out that December next year, we are going to have a global conference dedicated to both human and veterinary vaccines batch release strategy and how non-animal testing innovation is actually entering into the release strategy of those products. And as I said, if you have any questions, feel free to get in touch with me. So thank you very much for being with us today. And I would like to leave the floor to the first speakers, that is Laura Martinez from Virbac, that she's going to give us some information about the VICH new guidance on this topic. So Laura, the floor is yours. Thank you for being with us today.
Thank you, Laura. So hello, everyone. So as Laura said, I'm Technical Regulatory Affairs Manager in Virbac, so company in south of France. And I am working specifically in a 3R group for development of non-animal methods. And here today, I'm here to represent VICH subgroup. So I will trying to share my presentation, and that's where it's not working anymore for sure. Sorry, here we are. Okay. Laura, please, is it possible? Can you see my screen?
And all working well. Thank you.
Working well. Thank you. So today I'm here as representative of VICH subgroup in order to update you on the VICH work about transition to non-animal based veterinary batch release testing. So as you may know, there are already VICH guidelines existing for waiving of target animal batch safety test or laboratory animal batch safety test, so safety aspect. And so today we will focus on potency. So first of all, what is VICH? So VICH is the Internal Cooperation on Harmonization of Technical Requirements for Registration of Veterinary Medicinal Products. So you have 3 founding members, US, EU, and Japan. You have standing members, you have observers. And so WOHA is an associate member, and the secretariat is held by HealthforAnimals. So it's important to understand the role of VICH. So VICH is really here to harmonize the technical requirements for the data necessary for registration. So the idea is to provide a baseline of technical requirements and set of data, which will give confidence in the products to authorities. So you have VICH guidelines on study and testing methodologies or post-marketing safety monitoring. And it is absolutely not the role of VICH to substitute to authorities, okay? So it is a role of national competent authorities and governments to assess the data and to grant the marketing authorization. So we're talking today about the batch potency test. So what is it? It is a requirement for the batch release of veterinary vaccines. The idea is to ensure that a batch will provide the adequate level of protection from its release until its end of shelf life. So here you have the 3 different possibilities for batch potency tests. First of all, you have the in vivo test. So as an example, you have the hamster test for leptospira vaccine, where you vaccinate hamsters. You have control non-vaccinated hamsters and you challenge them. Then you have an in-between, so an ex vivo test where you vaccinate mice, as an example, for the rabies potency test. And you have controls and you understand if you have an adequate protection with the antibodies level. And finally, you have the in vitro test. And as an example, you have ELISA testing for the leptospira vaccine. So why are we pushing towards these in vitro methods? So first of all, for sure, you have the ethical reasons. So the idea is really to reduce the use of animals and to replace the use of animals in these batch release testings. This is a real demand also from the authorities, from some authorities at least, and from the public opinion also. And it's really a duty for the animal health industry also. Second, you have the removal of the animal variability. So you have in vitro testing that in some cases can be more accurate than the in vivo testing. And you have also industrial pros as, for example, a shorter testing period. Nevertheless, you have many challenges to face. First of all, during the development of the techniques, because it takes time, you have to identify specific regions, references, standards, technologies, and you have to set up specifications. And the idea here is to switch from in vivo to in vitro. So the idea is not to retest on animals anymore, to set up these specifications or less as possible. Then you have challenge in the implementation because the test should be transferable in a GMP environment. And finally, you have regulatory constraints in recognition or acceptance and implementation in the global regulatory setting. And this is a huge issue for the industries because they sometimes have to perform the both testings in parallel, in vivo and in vitro methods for some specific countries. Nevertheless, the regulatory acceptance has increased. So here you have a few non-exhaustive examples about the European Pharmacopoeia 5.2.14 about the substitution of in vivo methods to in vitro methods. You have laws in the US, also 9 CFR or memoranda. You have an Indian Pharmacopoeia 2.10.20, which is quite the same as European Pharmacopoeia 5.2.14, but with some differences. You have some things also in the Japanese law. And also you have collaborative studies between the industries and implementation in some specific pharmacopoeias worldwide. But nevertheless, we need to go further. So the concept paper was adopted in January 2024 by the VICH Steering Committee. And the mandate was to review the information which is currently in the European Pharmacopoeia chapter 5.2.14 and to review also over global guidance as well as the ICH, so the human work in this area, and to build a similar guideline to be assessed by the biological expert working group from the VICH. So the batch potency test subgroup was established and the first video was held in May 2024. So within this subgroup, you have representatives of regulatory bodies worldwide and also representatives of different animal health industries. And so the objective and scope of this guideline is, as I said, to provide a set of harmonized technical recommendations for this transition to in vitro methods for this batch potency test. In particular, we have to consider when there is no direct correlation between in vivo and in vitro assays. So as an example, when the measurements units are not comparable. But this is really discussions, open to discussions within the subgroup to work on this topic. And then the scope is for vaccines, and out of scope, we have live vaccines, because for live vaccines in a majority of countries worldwide, the in vitro methods, so I mean the titration test is already accepted, or any product that currently uses an in vitro potency test. So a draft of the guideline is established, but we are now the subgroup, we are really to go deeper into the details. So here are some examples of the major items we have to work on. So first of all, we have to ensure that there is no contraindication with local requirements, because as I said, the VICH cannot substitute to the local assessments. We will not include any details on the validation of the methods because they are already provided in other guidelines, whether in VICH guidelines for some products or in local guidelines. We need to emphasize on the antigen content and its link to biological functionality. Later on in this webinar, you will have the example of the rabies potency test. So you will see the test target in this case is really well known. But we can have over vaccine, as example, complex bacterial vaccines, where it could be really difficult to understand the relevant test targets. Then we will also discuss about the fact that we could use multiple assays in spite of a unique one, as for example, ELISA for the leptospira vaccines. Because, so as an example for these complex vaccines, it could be useful to have multiple assays. Then we are discussing also about the testings to assess the adjuvants. Why? Because when you're testing in vivo, you are testing the total vaccine, including the antigen and the adjuvant. Whereas when you have an in vitro methods, you target the antigen. So you need to understand the role of the adjuvant. And the test should discriminate subpotent batches. So once again, we need to work more on the definitions of subpotent batch or under-formulated batch or whatever we call. So there is no specific timelines for this process. So we have rounds of comments which involve all the organization and all the members. So I thank everyone for their work on that, because really the process goes smoothly and everyone is really involved, and that's really great. And we have video meetings every 4 to 6 months. And so the last one was yesterday. So you will see later on examples of success without this guideline, but we really think that with this VICH guideline, we will go further in the acceptance of in vitro testings. Thank you for your attention.
Thank you very much, Laura. Just since we have few minutes, I would like to give you a quick question, if it's okay with you. So what do you think that's would be the biggest complexity in implementing or transitioning these guidelines in local regulations? So VICH has this role really to facilitate some of those concepts to become a part of national or regional regulations. So, but what do you think that might be the biggest complexity to see this transition happening in the upcoming future?
But we are really working on this guideline in order to improve its acceptability in the different regulations. So as you know, you can have different mindsets between the Europe or between the US or between Japan or other countries on, as example, the testing for adjuvants, as I said, is not something which is requested in some regulations, whereas it is absolutely mandatory in others. So that is where we need from now to understand these differences and to work on this guideline in order to be, let's say, as precise enough to have this understanding from these different authorities, and in the meantime to be in a gray zone so it can be implemented without affecting the current regulation, and it's a real issue, and that is why now we are going we have, as I said, this draft of guideline, and now we are going a little bit a step forward. Because now we have really these bullet points, and now we go a step forward in order to understand the concepts behind these bullet points, in order to discuss maybe by subgroups. I don't still know how we are going to do that, so that the acceptability could be the best worldwide, because the idea is not that same as for target animal batch safety test guideline. As an example, you have additional requirements in some countries. We will be some additional layouts on the above the guideline, you know. So yeah.
You, Laura, thank you very much. So,
Okay.
thank you. I would actually move to our next presentation because it's actually coming through very nicely because Dean Smith from Health Canada is actually going to give you a little bit more details about what is the background about the 5 to 14 chapter that also Laura mentioned. So thank you, Dean, for joining. The floor is yours. Feel free to
Thanks.
start sharing your presentation. And
Okay,
while you
great.
do that, I would like to remember our attendees to put their comments or questions into the chat box or into the Q&A session into the Zoom window. So thank you. Dean, your presentation is coming up.
There we go. So first, Laura, thank you very much for the invitation to speak today and your colleagues, and hello to you all. As Laura mentioned, I'm from Health Canada, and I'm sort of representing a team view from our perspective, but even a larger team view from our work with European Pharmacopoeia and our work on, as Laura mentioned, 5, 2, and 4. So I will not mention 3Rs too much, but I will also agree that the humane use of animals is central, whether we're moving towards in vitro testing, but we still have ongoing roles with animal testing as well. And we are going to be with animal testing for many years for preclinical work on the human vaccine side, and of course, for forever in the veterinary side, as they are the target species. So, essentially, if you're not using humane practices in your work for your assay assessment, then you're not going to be getting reliable data. But for us, the main drive at Health Canada and our work with EDQM has been just to get to more appropriate in vitro assays, assuming they're fit for purpose and designed to be that way, because we've found very early in our respective careers with my colleagues at Health Canada and elsewhere that the in vivo assays have really been very substandard compared to what we can achieve. And this talk will hopefully reinforce that message for you. So first, I'll acknowledge the very large group of people involved in 5214, this general chapter in European Pharmacopoeia that was implemented in 2018. As you can see, there's a large team of direct participants in this, as well as people outside of the working group itself and outside of EDQM. This was implemented in 2018, as I mentioned. It was initiated in 2012, and it's really a 2-page general document, but it's been central to what's happened in changes, as you'll see through the talk. So an overview is to explore the barriers to the transition from in vivo to in vitro assays for key legacy public health vaccines on the human side, and of course on the veterinary side as well, including this. And the barrier here is the stories that we, because I'm a regulator and you're a regulator, we regulators believe that create a negative feedback loop for industry. And what I mean by that is if regulators believe that in vivo assays are essential to maintain safety of vaccines, and they are perceived as non-supportive of change of these assays, industry will simply not invest in innovation and new assays, and therefore no change can occur. So if we're interested in better science and better assays, the path forward is for a more science-based, less fear-driven, risk-aware mindset. And European Pharmacopoeia 5214 approaches support that. So the key message here is that we have this multi-decade dilemma with the transition from in vivo assays to in vitro assays. And that dilemma is resolvable through a data-driven critical thinking process by regulators as well as by industries to develop stories that better reflect reality to eliminate this negative feedback loop. But it's particularly important that regulators get this story because industry understands this, and I'll highlight some as we go along. Okay, so we have these shared goals, and these shared goals are both on the industry side and the regulatory side. We all, of course, want quality and safe, effective vaccines, but we also need those vaccines to get to market and be available. If they are tied up in regulatory processes, they simply can't be available for use, and that's true on the veterinary side as well as the human side. And of course, we've all unfortunately experienced this rather horrific pandemic recently, and getting vaccines to market was critical, and efficient strategies to do so was central. And you'll see that relates directly to this talk as well. So what we need is innovative thinking evidence-based decision-making to support manufacturing and testing strategies that lead to global regulatory harmonization and convergence. And this is central because it's already been mentioned by the previous speaker that, if we see advances in Europe or North America with movements to, in vitro testing, fit for purpose in vitro testing, and that doesn't take place in other regions of the world, that means a manufacturer who has invested and brought forward a better assay system, which is more effective, now has to run 2 sets of assays, which has doubled their work. So it's really critical for us as regulators to work together to move this forward in a more sensible path. And so the reality of in vitro testing versus in vivo testing is very evident in modern-based vaccines. We have a large number of products for decades now that do not use in vivo assays for their quality control. I remember here we're talking about quality control, not proof of concept work in early trials, et cetera. But we're talking about the final quality control of products. So examples here involve the human papillomavirus vaccines. These are recombinant viral-like particles plus an adjuvant, and they are controlled with physical chemical methods and ELISA. These have been very successful vaccines in the human field. Then we have the meningococcal and pneumococcal bacterial conjugate vaccines, again, highly successful products, defined polysaccharides conjugated to carrier proteins, and they are controlled specifically with physical chemical methods, no animal methods involved whatsoever, and not even a bioassay. EMA and North American authorized COVID vaccines exclusively used in vitro QC control strategies, regardless of their platform, whether that was mRNA, subunit protein vaccines, or viral vector. Animal use was restricted to proof of concept in preclinical studies, so nothing was involved in terms of animal QC work for these vaccines, and they were rapidly brought to market and kept on the market and extremely well controlled. So once upon a time, we all relied on only final release lot testing. That was the long distant days of vaccine. Now, of course, as you know, we test products throughout their manufacture. And if you're dealing with a large multi-antigen conjugate vaccine, with maybe 12, 15, 20, 21 valencies, there might be 300 or more tests involved in the production of that product, and we do that throughout the beginning. And of course, we're very conscious of starting materials right through the release testing. And so we have a whole battery of tests specific to each stage of that development, and we're controlling this all the way along. And I'll just step back and look at some of the problem areas that drove the development of 5214, which was that when I took over the rabies products in Health Canada when I joined in 2002, what I quickly learned was that all these rabies vaccines we had in our market, and there were 2 at the time, they were actually manufactured and formulated using in vitro assays, but we as regulators required them still to go through what's now referred to as the NIH animal potency assay, which we'll talk about a few times today. And so while they were actually formulated and controlled with in vitro assay, we still required this animal assay as though it were something, some golden measure of quality that we actually knew was already in place. It was a very frustrating process for me. We actually stopped doing that test almost immediately and relied on the in vitro assays, although of course we always kept collecting this, and we'll come back to rabies a bit later. So typically, as I've already mentioned, conjugate bacterial vaccines, their label claim is not, the potency is considered as certain number of micrograms per milligram of defined components, such as specific polysaccharides, or plus the physical chemical assays around the adjuvant itself. And that's an entirely separate conversation, but it's important to keep track of that. We can monitor adjuvant quality through physical chemical methods, again, not requiring animal assays, once we understand the components of those vaccines and what's critical to make them function. So in spite of all of that, when 5214 was initiated in 2012, it was done because of the massive resistance within the regulatory community to seeing these changes come forward. So we clearly had lots of ways to do this, but yet the rabies NIH test, for example, had been going for 40 years. And there have been many attempts to bring in alternative assays, the serial radioimmunodiffusion assays, the SRID, in vitro neutralization assays, stability indicating glycoprotein ELISAs, but no implementation of alternate methods. And in fact, 2 failed WHO collaborative studies to do so, not because of the new assays, but because of the variability of the existing assay. So the abnormal toxicity or general safety test or innocuity test, for 20 years, Paul Ehrlich colleagues in Germany have been trying to delete that test because it had lost its scientific relevance, but nonetheless, it was still very much part of the pharmacopoeia in Europe as well as in North America and other parts of the world. There's an assay which I'll introduce you to at the very end as a bit of a case study. That's the pertussis histamine sensitization test, or HISS. It had been on the market, and many assays, including those from colleagues at Health Canada long before I joined, developing alternates to this test, yet it was still very much in WHO guidelines and national guidelines. Toxoid irreversibility tests were still required, and that was after decades of stability testing in vaccines of these stable toxoids in all human vaccines, at least. Yet in vitro reversibility testing was still required. Again, the scientific rationale was lacking. Diphtheria and tetanus assays and pertussis products lacked progress in development of in vitro methods, and that is still ongoing now, but we now have many new alternatives coming in as again a function of activities of VAC2VAC and the European Pharmacopoeia 5214 together. Rabbit pyrogenicity is preferred by most authorities over a monocyte activation test. We won't touch on that very much today, but this is rapidly changing in Europe, and that'll be the rabbit pyrogenicity will be essentially off the European Pharmacopoeia very shortly. So the key message here is a scientific, less animal-centric mindset was needed at an international level to implement alternative in vitro assays required for world markets by vaccine manufacturers, and frankly for our public health needs. And that is the same for the veterinary products. So what are the key limitations that 5214 and Group 15 and Group 15V, the veterinary partner of that group, were trying to challenge as we went through? So it's already been mentioned that these assays, they are highly variable, and that has resulted in multiple failures of multi-center international collaborative studies requiring one-to-one comparison of products, not because of the more precise and consistent in vitro methods, but the variability in the, for example, the rabies NIH test, which made it impossible to get alignments in collaborative studies. Another key factor here, which is it's described in 5214, is that most in vivo assays predate the ICH Q2R1 for the human products or the VICH GL2 guidelines, yet they are considered validated since they are compendial assays. Hence, one-to-one comparisons are challenging or not possible because we don't even know the precision, reproducibility, and limits of detection, et cetera, for the animal assays. They are simply not established. And under EU conventions now, where there's great restrictions on use of animals, it would be unethical or not even feasible to obtain the animals to do those studies. So that makes those transitions very, very difficult. Another part here is that while properly established in vivo methods now have the potential to measure complex functional responses for demonstrating proof of concept, they do not predict the response in the target population, meaning that's in the human population in this case, but they are merely highly variable bioassays. Of course, if you're working in your target species in a veterinary context, that's a different story, but still it's a highly variable assay, and if you have more precise means of doing so, of determining potency or safety, you should be using those tests and not an animal test. So fit for purpose in vitro alternative assay Q studies using one or more new methods will likely assess the same quality attribute differently. Hence, the expectation you have a one-to-one correlation between these two methods is not really scientifically justified in all cases. But yet a fit for purpose in vitro assay can and must provide at least the same confidence regarding the control of the key quality attribute, and case studies have supported that expectation. I'll mention those briefly a little bit later. So there are some key sort of words to keep in mind in this discussion. So in a European context within the European Pharmacopoeia, terms like replacement and now substitution have specific regulatory meaning. Replacement involves the one-to-one comparison of an established correlation between two methods, e.g. in vitro to in vitro to in vivo, excuse me, or vice versa, or even in vivo to in vivo if you're proposing a new method. Substitution, on the other hand, this is what is specific to 5214, this general chapter in European Pharmacopoeia, is to facilitate the implementation of in vitro methods as substitutes for existing in vivo methods in the case where one-to-one assay comparison is not appropriate for reasons unrelated to the suitability of one or more of the in vitro methods. So the assays, of course, must be fit for purpose, and we demonstrated that. And again, here we're talking about really on the human side, an ICH or VICH approach, that standard assay implementation demonstrating fit for purpose and consistency depending on its application. And stability indicating is a term I'll mention a few times, and it's fairly well-known concept, and I've listed a WHO TRS here where that is defined, but these are quality parameters, direct or indirect indicators of vaccine efficacy or safety that are sensitive to storage conditions. These parameters are used in stability assessment, product quality throughout the shelf life, and determination of these parameters should result in a quantitative value and a detectable change, over storage condition. So if it's stability indicating, it tells us what the potential of that is likely to be in the clinic if it decreases or increases any of its specific parameters. So these concepts are basic to understanding how 5214 is implemented. So while the drafting group for 5214 was deliberating on how we could come up with a new proposal to go forward that would be useful in a regulatory context, there was a decision by the FDA to delete the general safety test, and that spurred a meeting in Europe organized by EPAA in 2015. And we had a few speakers, and these presentations became very useful in the discussion around 5214. So industry, in this case, Sanofi, stressed the resource drain with multiple versions of in vivo potency and safety assays for several DPT vaccines, month-long lot release delays with invalidity results and repeat testing, which typically would delay otherwise compliant lots to getting to more. Here we have assays meant to control quality actually resulting in vaccine shortages, and there are several examples of that. As I mentioned earlier, the German authority, Paul Ehrlich, presented very clear case for no scientific rationale to retain the so-called general safety test or the innocuity test, which was designed initially as a phenol test for tetanus antitoxins in the 1900s, and it lost its scientific relevance in the 1930s. When we developed chemical assays for that, but yet we just decided to keep going with that assay, and we called it the general safety test. The UK's National Control Laboratory, and actually it was Paul Stickings, it was Natural Institute of Biological Standards at the time. Now it's the MHRA. They presented ELISA results versus in vivo potency assays for diphtheria and tetanus products, demonstrating a higher sensitivity and improved stability indicating potential for the in vitro methods versus the in vivo methods. So this idea that somehow in vitro assays were giving us better control is essentially a myth, and this is one that the 5214 is meant to take down. The key GST general safety test and diphtheria tetanus in vivo potency conclusions from this EPA meeting were brought back to the 5214 discussion, and that data was very compelling and helped drive 5214 to its completion. It was a very long and complicated process, and all that's to say that the solution to that was that this joint work between industry and innovative regulators was central to the development of 5214. And VAC2VAC, which I'm not going to talk about, but will come up a few times today, I believe, which was a consortium of industry and academic and regulatory groups, was started essentially in parallel with 5214 process, but they were independent, and that was to overcome these barriers within the regulatory world. So what does the 5214 state? And these are literally quotations out of this European key regulatory document. So this is regulators speaking to regulators now. All QC methods should ensure comparability of the quality attributes between commercial batches and those batches originally found to be safe and efficacious in clinical studies for veterinary vaccines in the target species. So that's fairly straightforward. However, the inherent variability of in vivo assays can make them less suitable than appropriate As a result, it is essential to continually challenge the scientific value and relevance of these in vivo methods. Now, this is a regulatory document from a central agency in the world. The use of appropriate in vitro methods enhances the predictability of release of safe and effective vaccines for lot release. So, key message here is Group 15 and Group 15 move past the fear related to loss of animal assays. This was a result of an evidence-based discussion where long-standing beliefs, which I refer to as myths, were challenged and put aside. So key elements in this that I'll continue with: the primary focus of implementation for any proposed in vitro method within a QC system should be the scientific relevance of the in vitro assays and the control strategy. So scientific relevance over regulation. Keep that in mind. This is coming from a regulatory document. While the European Pharmacopoeia in vivo assay replacement with in vitro assays is typically achieved through multi-center collaborative trust, this should not be a prerequisite for alternate assay that works. That's something that you don't need to go to collaborative study. You should be able to assess that on its merits and go forward with that. And that's true in Europe as well. And this is being endorsed by the European Pharmacopoeia note. While it may be desirable to have assays that fit a wide class of products, this is not a requirement. And this is a unique thing about vaccines due to their long history. I won't go into the details, but we've often required manufacturers to come up with assays that work in a class of products. That is not done in biotherapeutics. A manufacturer of biotherapeutics only needs to have a control strategy that works for their product, and we need to think a little bit more like that in the vaccine side, in our opinion. In some cases, an existing in vivo method may need to be substituted by more than one in vitro method to characterize critical quality attributes as measured by the existing test, meaning a simple, or not a simple, but a complex and complicated in vivo test will give you a single potency readout, but to capture the relevant CQAs for that same vaccine, you may need multiple tests to do that. And then approaches for potency assays, design stability indicating assays, combinations of alternative methods to capture this. General fit for purpose, as I mentioned. Safety assays would consider different types of assays presented for specific toxicity, molecular consistency or next generation sequencing versus neurovirulence testing, and detection of viral extraneous agents by other molecular majors such as NGS. And that is now about to be amended in this guidance as well. So I have several lists here. I just want to point out a few key ones. After 5214 was implemented, we saw very rapid change at the European Pharmacopoeia. And eventually in 2023, and I'll give you a quick example of this in a moment, the rabies test was replaced with one product in Europe. The abnormal toxicity test was removed. The histamine sensitization test was removed. You see a lot of these other toxoid reversibility tests were removed. And I'll move on here to my other list. We saw these adventitious agent tests being replaced with other methods, other approaches. There are many efforts going on now in Europe and North America on that. And of course, as I mentioned, the COVID vaccines didn't use these. And the sub 5214 has been so successful that the WHO is now developing their own similar document, a technical report series document that will capture hopefully the key elements of what 5214 is. So hired in 2000, but still persists in many jurisdictions. This is a test that was introduced in 1981. The basis of the test is mice are naturally resistant to histamine, but the exposure to pertussis, pertussis decreases by upper 300-fold. So it's not even testing the toxin directly, but it's an odd relationship. However, what you need to understand about pertussis toxin, it's unlike diphtheria and tetanus toxins, it's not a very toxic material. People who suffer from exposure to, or not suffer from, but who are exposed to pertussis toxin only suffer a mild wasting condition over many years, and existing licensed products, even in many parts of the world, have 100 to 350 units of pertussis toxin in those products, and that actually makes it a better vaccine, surprisingly. And there is a Chinese hamster ovary cell assay that for decades has been used by industry, which determined by the European Pharmacopoeia was we had an in vitro test already in place. It was widely used by industry, and this histamine sensitization test was essentially redundant to the test requirements and was deleted. A quick example on the rabies protein here. I'm just going to quickly go through this. We're comparing here ELISA results with this new Sanofi ELISA versus the NIH test. These are two test conditions. The same lots 1 and 2, are being tested. And if we go over to this central column here, the intact vaccine, they all are shown to be compliant. But notice NIH test. And most importantly, when you then heat treat these products, you can lose all of their potency, which is what you'd expect to see. And that's true of both the NIH test and the ELISA. But now if you spike these materials with valid product, you can see that you're still not recovering the material in the ELISA here in the far right column, the two top results, whereas spiked results with the NIH test still pass the test. So if that's a gold standard test, it has clearly failed. And this is being shown that to highlight the fact that once we make a transition to a better assay, i.e. this ELISA, we should not be requiring manufacturers to go back and reuse these tests, which they lose the specific expertise to conduct, because there's no scientific rationale for doing so. We have better methods that control these products with these in vitro tests if they're properly designed, and that's what we should stay with. So next steps, as for European 5214, Group 15 and Group 15V will continue to examine the scientific rationale for existing in vivo potency assays and safety assays with a more informed science-based approach with vitro methods for vaccine characterization and process control and QC assays as substitutes for in vivo methods, and global acceptance of appropriately developed fit-for-purpose in vitro QC control strategies is essential given the world market for vaccines. And note again that the COVID vaccines that sustained us quite well during the pandemic, all did not use, at least those approved in North America and Europe, did not involve animal assays and wrong QC strategies. So European Pharmacopoeia 5214 approaches will continue to accelerate the transition from in vivo to in vitro QC methods and for more effective, robust alternative control assays. So I'll stop there. Thank you so much. Happy to answer any questions.
Thank you, Dina. We actually have one question from the Q&A. So I'm just going to read it, but you can open up also on your side. So older veterinary vaccines are highly effective, but their critical quality attributes might not be fully understood. An in vivo potency assay, vaccination and challenge serves as a de facto confirmation of the potency. What do you recommend to transition towards in vitro potency test for a vaccine that, although potentially crude, demonstrate high efficacy, but lacks a comprehensive understanding of its critical quality attributes?
Yeah, this is a very important question, Laura. Thank you. And this is one of the advantage of the 5214, or sorry, the Vac2Vac consortium, is that for all of these legacy products on the human side, and now you're talking about legacy products essentially on the veterinary side, is that to invest in products which are generally low profit yielding medical products or veterinary products in this case, working together with companies who are in a similar space with a similar type of product to begin to understand, you need to characterize these products. You can't develop alternate assays unless you do the characterization you're essentially alluding to that allows you to understand those CQAs. So this is the black box of these legacy vaccines, and that was the wisdom of the Vac2Vac consortium, is that it combined resources of industries in similar sectors. And initially it was Sanofi and GSK. It eventually included Pfizer, and on the veterinary side, Kat can speak to that more wisely than I can. Is that a large group of effort? A large amount of effort was shared by industry to characterize these products and develop new methods. That's really the only alternative to do that. I appreciate that's not available to all specific individual manufacturers, but you can learn what was useful for a process like Vac2Vac. And again, those manufacturers who've worked in that space, and I'm sure, although they all are competitors, at least out of Vac2Vac, there were many publications that came out of that work, and I highly recommend other manufacturers to be looking at those publications, and that will give them some ideas of what types of strategies would be most appropriate for them.
Thank you, Dean. Please, Kat, jump in.
Just to add to that, I mean, I think one of the things that comes out of Vac2Vac, I agree with Dean, you need to understand the product and the process, and obviously you haven't done that through development because these vaccines were developed 40, 50 years ago. But what you do have is you do have a may not have defined critical quality attributes in the way you would do now. What you do have is a long history of consistency information, of understanding what your routine process parameters look like, what the ranges and everything look like. So you can take a consistency approach with these old products, and it's very much how we approach setting specifications for a lot of these old products, is looking at consistency data. So we're trying to ensure that each batch that's being released with the in vitro tests is consistent with, rather than necessarily showing it's consistent with the batch that was used in your efficacy study, you're looking to ensure that it's consistent with the historical information, that's shown that batches produced with within those parameters will pass the potency test.
Yeah,
So,
Ken, I agree with you completely, but can I interrupt for a sec? You're still going to need to understand what the CQAs are going to be, even in a consistency method. But I agree with you that in a consistency method with these legacy, it is an easier task than starting with novel products because you at least have that consistency data there to work with. But you'll still need to find something that is stability indicating relevant to the efficacy of the product.
Yes, absolutely. And I think that's where we come in, and you'll see with some of the rabies examples, the ways in which we've gone about, I guess, that we haven't defined them as CQAs in that context, but ensuring that the methods you're using are going to ensure that they will pick up batches that are subpotent or subformulated. So we're not talking about critical quality attributes all the way through the manufacturing process. Rather, we're talking about critical quality attributes in the context of potency. So I think we have to be a little careful with the terminology of critical quality attributes, because those now, in a modern manufacturing sense, GMP and everything else we're doing now, apply slightly differently all the way across the manufacturing process. Whereas here, what we're talking about is ensuring that the tests we're going to use to replace the in vivo are capable with the product, in the same way that the current in vivo tests will pick up what is perceived as any lack of potency. I'll put it that way.
I agree completely, Kat. And in fact, what you'll find is with an appropriate in vitro test, you're going to more likely pick up those differences, and that's something else that needs to be factored in.
And that's actually a bigger challenge, Dean. Actually, is that as we're starting to try and set specifications with the in vitro tests, you're starting to have to narrow and refine your specifications for these older products, and look to, in many cases, we have to move to ensuring we're formulating an antigen content tests in addition to testing with them, because if you rely on previously pre-inactivation titer, etc., it's not predictive. And that's one of the things that we all understand, that actually, as we move towards these in vitro tests, they don't just give us better confidence in the release testing themselves, they give us better confidence in our manufacturing consistency.
Agreed. I'm sorry. One last thought, if I can leave you with, I know I'm cutting your time here, is it just to regulators, is as manufacturers are moving in that transition from a highly variable in vivo assay to a more precise quality control strategy that Kat's describing, you shouldn't be trying to narrow them down to much tighter control strategies that they were working within these highly variable assays. You should try to be capturing the window that was relevant to the product's performance in with the highly variable assays and start there. Lots down, you'll be taking perfectly valid lots off the market, and that would not be appropriate for your animal husbandry areas or the manufacturers themselves, of course. But even from the quality perspective, from the public health side or the animal health side, being generous with those specifications within the range of experience and your consistency, as Kat's talking about, is really rather essential. I'll get off now. Thanks so much.
Dean, all of that says is the importance of it being data driven. And
absolutely,
using the data to define those specifications.
Agreed. Bye-bye.
Thank you, Dean. Thank you, Kat. I would move to actually our next presentation, because, of course, we have been talking a lot about the Vac2Vac project. And we have here Carmen Gumbek from the International Lab for Biological Standardization, who is actually giving you a little bit of background about this important project that, although it is over since a couple of years, but its impact is still having us discussing here right now in terms of outcomes or still activities that still need to be moved ahead. So Carmen, thank you very much. The floor is yours.
Thank you, Laura, and thank you giving me the chance to explain the role of VAC2VAC in all these things. As Laura and Dean have already indicated, and I think the speakers who will come after me will say the same. The development and the efforts to replace in vivo tests by in vitro tests does not come out of the blue in the last few years. This was really a long-term work with more or less success and more or less acceptance by regulators, manufacturers, etc., etc. And therefore, some people were really, to say it so, fed up and say, now we need a joint approach. And therefore, VAC2VAC was implemented. This is was really an effort to bring all people together and push the development of in vivo tests or non-animal tests forward. And therefore, this VAC2VAC project, next slide, please. It was in Horizon 2020 a project, and we started in 2015, ended up in 2021. Normally, it's a 5-year project, but with this lockdown in the Corona times, we have an addition of a year prolongation. Next slide, please. Yeah, there were 21 participants, 15 public partners, 3 FPR companies, 3 Health4Animal companies. So everybody was together. The budget was high for our purposes. This was nearly 50-50 funding for the public, for the OMCL's public partners, and the money the FPR partners added to this public money. So next slide, please. Here you can see an overview, and what to me was one of the good things. Can you see my file here? The national reference laboratories have a very close connections to the regulatory agencies. Sometimes these people were sitting door to door, which makes communication easier and helps pushing all the results forwards to regulatory provisions. Next slide, please. Here we had this One Health approach for the vaccines for humans and animals have the same challenges. And Kat Sterling will talk about rabies later on. Oops. And this is really a test which fits for human and veterinary vaccines. And so far, it's really something which is benefit to humans and animals in both ways. The cross-collaboration between the public and the private partners was extremely beneficial. And so we can really say that the VAC2VAC project acts as fosters the whole approach towards non-animal testing and really works as a catalysator to push these approaches forward. You have already heard from Laura the update from VICH, and this is was not possible without the acceleration and the more open-minded approach, which was really pushed, for example, by this VAC2VAC project. Can I have the next slide, please? Yeah, this is really the reality, the state of the art at that time. How many animals were used worldwide for testing? Then Dean had already talked about it, the variability of animal potency tests. And there are a lot of factors, the animals itself, the lab, the handling, the people who work with it, and the products. And so far, I am so far that I say I never try trust an animal potency test. This is my lesson learned from VAC to VAC. So then we have really difficulties to control the in vivo assays, for there are a lot of variables inside. You cannot predict the efficacy in the target species. It is time consuming. This is really a negative impact to the availability of vaccines, especially in emerging situation or when there is high disease pressure and vaccines are urgently needed. These animal tests are costly, and as I said, it hampers the vaccine availability. So, and the vitro alternatives, Dean has already said this, very precise. They are more consistent, more reliable. You know what you are test. You can identify the parameters you wanted to control to ensure the consistent production. Next slide, please. Yeah, oops. In vivo. Within the project, there was a very important publication from Stolpers et al. They have analyzed the in vitro potency release assays for DTaP vaccines. And these tests were pharmacopoeia tests. They were so-called standardized. They were confirmed in collaborative studies. And even there, a large degree of variability could be demonstrated. And so far, whoever wants to see how high the variety of an animal test can be, please have a look at this publication, for this is really an excellent example to demonstrate the difficulties for in vivo tests. So I have already said the critical quality attributes can be better defined in vitro tests. And from VAC to VAC, we had a lot of achievements, the DTaP changes, the TBEV ELISA, veterinary rabies, and the Clostridia chauvoii ELISA. Dean has already indicated which changes in the European Pharmacopoeia had been made, and all these test results have helped to change the Pharmacopoeia. Next time, slide, please. Yeah, the abnormal toxicity testing was already skipped in most of the Pharmacopoeia provisions, and therefore it was not a subject of the VAC to VAC project, but should really mentioned here as a very big success to reduce unnecessary or delete unnecessary animal trials. Next slide, please. Yeah, the barriers and the fee factor. Dean has already talked about it. It's always the problem to persuade regulators if a manufacturer comes across with a new test, and vice versa. In so far, VAC to VAC was really a good approach, as here both parties work together and there was no need to persuade each other. It was only the need to develop the test together and then check if it really works. The problem we have is sometimes a political pressure. It was told by me to some of my regulator colleagues years ago, whenever a vaccine fails, then the politics come around and was it already tested, was still tested in animals before and whatever. And when we change to pure in vitro testing, then this may be sometimes a hurdle to overcome. Okay, everything, whatever is new, people have fear. This is the normal protection of manhood all over the world, but we should not be too shy to look at novel developments. And therefore, we have to approach to understand which barriers we have to overcome, and then come over with data, as in the discussion between Kat and Dean was already mentioned, provide data and then persuade everybody who does not really understand the problem. And what is also a part of the new approach is that the in-process controls will become more and more important. So the old vaccine production is you start with a vaccine and at the very end you tested it. With the modern methods, you have more in-process controls to check if the production process is stable and reproducible and consistent. This is the new approach, and this helps maybe to persuade the people that we have to change the final product testing. A wonderful example for non-animal tested vaccines are the COVID vaccines. Their animals are only used for preclinical development. And I was told, sorry, I'm retired since some years, I was told from my colleagues in the human and veterinary field that the new and modern vaccines, which are licensed in the meantime, mostly are licensed without animal test at the final product. So can I have the next slide? I think I'm nearly through. Yeah. The way forward, this is really something that the GMP production is something which is more and more implemented globally. The in-process controls are increased and will ensure consistent production as well. And this allows as well to detect non-conforming batches much better than for the in vivo test. And the big variability of in vivo tests can be excluded. Kat has already said that replacing in vivo test by an in vitro test for long-term licensed vaccines is based on the look at the historical data. And what helps us as well, here again, we come with the problem or the point of availability of vaccines when these in vitro tests can be performed much easier in a lot of labs. And so far, the comparison and the acceptance of data should be much higher than for in vivo tests, which may differ from laboratory to laboratory. Yeah. The whole approach enhances the availability of vaccines, and as all together work, the regulators, the OMCLs, and the manufacturers and academy people, then I think we are on a very good way to delete the last in vivo tests, the remaining in vivo tests, which are still there. We have a lot of references. They are here, and I think the slides will be available after this webinar. And I think I am on my end here, and thank you for your attention, and hope I could have illustrated a little bit the background for the presentations we have today and the developments which are really undertaken at the moment. Thank you.
Thank you. Thank you very much, Carmen. In the interest of time, I would probably move to the next session because it's going to be a big one. I would like to welcome everyone to type their questions or comments into the Q&A box so that we will try to see whether we can answer to your question during the next section or in written. So it's a pleasure now to move to another important session of this webinar that is dedicated to the rabies vaccine. It has been actually mentioned more than once as an important case study today. So I would like to welcome the group of speakers that today are going to talk about this topic. I will bring up the presentation in a second. So we will start with Kat Stealing from Zoetis, following by Benjamin Attas from Boehringer Ingelheim, then Peter Peppers from MSD Animal Health, and then last but not least from Ignacio Gisbert from Zoetis. Kat, the floor is yours.
Thank you, Laura. Do you want to put it up full screen? I'm just going to give you a bit of an introduction and a conclusion at the end here. It's Benjamin, Peter, and Ignacio who are going to give you the core of this. So what we're going to talk about here, and this goes to what's already been presented a lot by both Dean and Carmen and Laura. What you'll see through all of this is a lot of common themes that go back to what Dean, Carmen, and Laura have already talked about in the way in which we approach this. And this is really showing you the industry success on the veterinary side in approval of rabies glycoprotein ELISAs for potency. So this is replacing either straight the NIH challenge test, or in some cases the interim step, which was the mouse serology test, which was developed previously by the Paul Ehrlich Institute. But in all cases, we're moving away from using any sort of animals. So if you go to the next one, Laura. So it's taken us probably more than 40 years of discussion, and I'm sure those online who've been involved in rabies testing know that both in the animal health industry and in the human health industry, we've been talking about trying to replace the NIH pharmacopoeia animal tests for rabies with an in vitro potency option. As has been mentioned already, we have had success in having changes made to the pharmacopoeia monographs for rabies that specifically mentioned the use of immunological methods, in this case specifically the glycoprotein content ELISAs. And it's been a long-term collaboration, working very closely with regulatory agencies, official medicines control laboratories. So in Europe, OMCLs, they are the bodies that do the retesting. So for those of you who are not aware, generally speaking, in Europe, we operate on an administrative batch release approach, but we do have, which means we're just reviewing batch protocols, but we do have a specific list of tests where there is retesting by the authorities for potency, and rabies is one of them. So rabies is one of them that's a little bit more complicated because we need to ensure that the medicines control laboratories can also do those potency release tests. And although it's taken a long time, we have finally got there. And following EU approvals, companies have also succeeded in replacing the in vivo test in many international markets. And both Benjamin, Peter, and Ignacio will show you in their presentations where we've had success in this globally. So what we're going to do now is we're going to go through each company's. I've chosen the order it's historical, so it's who got there first. So BI got there first, when still was Merial, I think, Benjamin, then MSD, and then Zoetis. And what you'll see is we have 3 different methods. So all of them are glycoprotein ELISA methods, or immunological methods, but they're all different. They're all different because the vaccine strains are different, the vaccine formulations are different. But as I said, you'll see common themes throughout that are consistent with both the EP monograph 5214 that Dean talked about, the principles of the consistency approach, which I talked about earlier, and where Laura and the VICH working group are working towards, and the same principles that we hope to be embedded in the new VICH monograph. So Laura, if you go to the next one, I think it's pretty much over to Benjamin.
Thank you, Kat, and hi everyone. I'm Benjamin Atta. I'm a technical regulatory affairs manager, and I'm member of the Animal Health Europe 3R task force. And I'm going to present you how we handled and are still handling actually our switch to in vitro potency test for our rabies vaccine. I will give you a bit of context. We started actually more than 10 years ago, and then we will look at some specificity of our ELISA, and then explaining how we have set our specification at both release but also end of shelf life. And at the end, we will look at the regulatory aspect, how we handle the variation in the various countries. So in terms of context, our main target was rabies vaccine, which is our monovalent vaccines, which is inactivated and adjuvanted with aluminum hydroxide. And actually, we are marketing this product since the early 80s. And for years, we have performed the NIH to release our batches, which was, of course, the standard at that time. We know the limits of this NIH test. There is, of course, the 3R concern, which is the one why we are meeting all together. But there is also another aspect which is quite important and which has already been raised by people talking before us, is that this test remains only a semi-quantitative one. So moving to alternative methods may not only improve the 3R aspect, but also the monitoring of better batch-to-batch consistency. Next slide, please. So, as said earlier, there was this first improvement at European level with a serological test developed by the Paul Ehrlich Institute and which was then tested by manufacturers and also official laboratory from authorities in the frame of a collaborative study driven by EDQM. And this was successful because then from 2012 or 2013, this test appeared as a possible alternative for manufacturers to release their batches of rabies vaccines. It was for sure an improvement from the animal welfare perspective, and in particular the suffering of animals. Still, it remained only a limited quantitative tools. This is why there was still an opportunity to develop full in vitro methods. And Merial at that time, now Boehringer Ingelheim, ended the development of the methods in 2015. And it was then published, and we have then deployed the regulatory variation from 2017. Interestingly, the package that supported our variation at that time was fully in line with the Pharmacopoeia monograph 5214, although it was still a draft at this stage. Slide, please. So now we are going to look a little bit more in details about the ELISA methods Boehringer has developed. So this method is a sandwich ELISA and with very well characterized antibodies. There was a lot of studies performed to make this characterization, and you have here two illustrations of those characterization studies. One is about temperature, so it's on the left side. You can see that you have two ways of performing culture of glycoprotein. In the dark gray, you have a culture at 37 degrees, and in light gray, a culture at 60 degrees. And you can easily see that while performing the culture at 37, you have still a detection of the glycoprotein even after 72 hours of culture. Whereas when you push your culture at 60 degrees Celsius, from 15 minutes, you already lose your signal and not have any signal afterwards, which is a first confirmation that our ELISA is not only targeting the good glycoprotein, but the glycoprotein in a certain conformation, which is sensible to temperature. Similarly, on the right side, you can see then some pH treatment to see also whether the acid or basic pH can influence the detection of the ELISA. And again, the figures are showing that the more the pH is running to acid, the more you're losing your signal in terms of glycoprotein. Interestingly, this is a reversible phenomenon. So when you are going back to a classical pH around 7 or 7.5, you have back your detection of signal, which is reflected in light gray. That is all to say that we have an ELISA targeting in its trimeric perfusion form the glycoprotein G, and we know that it is only in this conformation that it elicits the neutralizing antibody. So definitely a correlation and a characterization of the ELISA. I'm afraid we will not be able to reach this level of characterization in all our candidates for switching to an in vitro test. We have tested this test, and it was successfully demonstrated to be a quantifying test because it is able to quantify the glycoprotein at all stages of production, so not only upstream, downstream of the antigen, but also in a formulated vaccine environment. And it is also able to quantify this antigen during all the life cycle of the product. So not only at batch release, but it is also a stability indicating tool. And last but not least, it is able to target the antigen either in a classical and simple environment or in an adjuvanted one, like the one we have in Rabizin, which is adjuvanted with aluminium. We perform a full validation of the tool according to the ICH guideline 1 and 2. So at the end, it was definitely for us a good alternative for NIH. For the two reasons we already mentioned, 3R perspective, better batch-to-batch consistency monitoring, but also safety benefits for lab manipulating the strain, because it avoids also the manipulation of live rabies virus in the frame of challenges. And again, of course, for lead time to market, which is especially critical for rabies vaccine, which are subject to tender markets. Now, the way we established our limit of acceptance, we started first by establishing our release specification. And as said earlier, it was definitely based on batch-to-batch consistency data. We gathered the data of all batches released over a period of 3 years, and then analyzing them through statistical analysis and obtaining a mean value with standard deviation. And after that, we also tested the ability of the tool to detect subpotent batches, which is a requirement for batch release, of course, and for batch potency test more precisely. So we had already, because of the VICH validation, the data we obtained from the linearity validation, but they were also supplementing by tested experimental batches, which were voluntarily formulated at lower value than the standard one. And here you have the illustration with half a dose or a tenth of a dose. You can see that from the ELISA titer perspective, you have a good relation between the dose and the ELISA results obtained, whereas on the other hand, as a NIH testing, you have still a NIH signal positive, which in theory would have authorized the release of the batches, whereas we know already that the dose was not the one usually targeted at the standard. So again, illustrating the better monitoring of the batch-to-batch consistency. And at the end, we applied the statistical rule that the specification would be set at the mean value obtained from the batch-to-batch consistency and applying a plus or minus 2 standard deviation. So at the end, 2.45 is our specification for Rabizin. If we move now in the way we have established the end of shelf life specification, we have put in stability program 3 batches of Rabizin. So starting with a classical protocol and testing regularly the ELISA from T0 to T plus 3 months of the required shelf life, which is 3 years for Rabizin. And through a modelization of the mean loss in titer over time, it appears that our batches are losing 0.36 units over these 3 years. This was further confirmed by additional data. We picked several batches we were in their course of shelf life, so ranging from 19 months to 37 months. And this, the analysis of these further batches confirmed actually the first modelization, obtaining this time 0.34. So let's say exactly the same as the modelization. Based on this loss in titer over time, it was then possible, considering on the one hand the release specification, which was 2.45, and on the other hand the loss in titer through the time, to deduce our end of shelf life specification. And this is definitely an improvement, again, from consistency perspective, because we are now able to monitor the quantity of our antigen inside a formulated vaccine, which was definitely not possible within NIH. Next slide, please. So now a bit of information about the regulatory procedure. So we started first in Europe. The way was more paved than anywhere else in that sense. And we actually managed it in 2 steps. In 2017, we applied first for a switch to the ELISA method and introducing our batch release specification. The stability data I just showed you before were still ongoing at that stage. This is the reason why the first intent was not to also replace the stability monitoring and expressing the composition with this ELISA. It was done in a second run of variation in 2019. We have a registration in all Europe, but these are, as I showed you before, national marketing authorization, so well before the birth of MRP or any CP procedure, but we run it through a work-sharing procedure, and ANSES, so the French authorities, was our reference authorities to do that. Then we decided to run the variation also internationally. So I guess you know that in most of international countries, granting the marketing authorization in Europe is either a prerequisite or a facilitator to grant then authorization outside Europe. So this is why these variations outside Europe were launched afterwards. And we are still on our way. We have good completion in a majority of countries. Or the procedure may be still ongoing in some others. Interestingly, and this is my last message, you have a list of countries where it was not possible to make the registration of this ELISA test, either because authorities clearly told us don't even try, NIH cannot be substituted in our country, or we tried a submission, but unfortunately we failed. And you have here the list of countries, which is Argentina, Brazil, China, Ecuador, India, Nicaragua, Peru, Russia, Thailand. We will see, and I think it's from my colleague from Merck, that they were successful in some of the countries that I here mentioned as countries where we fell. So I think it's an optimistic message to end my presentation, that maybe we should try again in some of these countries and this time be successful.
Thank you. I think that there is a question for you, if it's okay that we quickly take it right now. Yeah. So it's from the Q&A, so you can open it and read it also while I'm reading to you. So rabies is one of those products that requires the use of BRP and a relative potency assay for release. How did you handle the decision not to use the BRP as a reference, and how challenging was it to convince the authorities of this new approach, which is disruptive at some point?
Mm-hmm. Yeah, these are clearly questions that we ask ourselves at the time of developing and implementing the test. We have used the BRP in all our development to see if what we saw in our vaccine was also visible in this BRP. Then it's also a mindset to switch from a relative potency test to an absolute one. So giving the chance not to look at a reference anymore, because we are looking to historical data consistency, as already mentioned by Dean and Katrina. Still, it's, I think, something that may be of interest for you. BRP was used at a certain start of using in routine this test in BI as an internal reference, not to compare the results, but to validate the session where we run the test. And then I guess now we don't perform it anymore, because again, you have to think that it's no more a relative potency, it's an absolutely one that can get rid of the comparison of any reference.
Please, Kat, jump in.
Yeah, no, I just wanted to call out the differences, and yet you'll actually see with these different presentations, there's slightly different approaches. So as Benjamin says, in this case, BI chose to set the specifications based on an ELISA value, an OD value. So you're not using a BRP reference. And in the examples where a BRP reference is being used, it's purely being used as a reference standard rather than any relative potency measure. And that's something that came up. There was a lot of discussion about that in the IABS meeting on references and international references. So it's changing the use of the way in which you're using the reference. So we're not trying to do with any of these methods, it's not relative potency. It's simply, if you are using the BRP, it's simply as a reference standard to get a value in international units, but not that necessarily that value in international units is necessarily a required threshold value, as has been the case historically. So there's different approaches here from the different companies, and I think you'll see that in the way in which it's been approached. So again, that's something different to what everybody's used to, particularly with rabies. I think, Laura, we're probably
Kat. We can go. Yeah,
next.
exactly.
Thank you, Benjamin.
Peter, the floor is yours. Thank you.
Okay, thank you very much to the organizing AFSA committee for inviting MSD Animal Health to present our journey, how to develop the in vitro potency test and how to license it. My name is Peter Pepels. I'm from MSD Animal Health Regulatory Affairs, located in Boxmeer in the Netherlands. Next slide, please, Laura. Before we go to the in-depth methods, a little bit of background information on our product. The product is Nobivac rabies and Nobivac rabies lepto, so it's also a multivalent product that we have in which the rabies is included. Nobivac rabies is for more than 3 decades already licensed, and already from the start of it, we formulated on a fixed amount of rabies glycoprotein, which is determined by an ELISA. Like Dean said, it was already we already had the ELISA in the early 90s implemented. And if you have such ELISA, it makes the transition to a in vitro potency test, let's say, relatively easy. The potency of our vaccine is at least 2 international units. We performed the mouse challenge test for 40 years, but we were very happy that in 2015, De Pey and Beate Kramer, and I also think that Heike Berensdorf was involved, who will later on provide a presentation, that they developed and validated a serology mouse test. We participated in a ring test, and then in 2015, the variation was approved, and it was already a tremendous step forward because we could reduce the number of mice with by a factor 10, and obviously no challenge was anymore needed. However, this serology test, it still uses animals, and also it uses live rabies vaccine in the lab, because you will do a virus neutralization test on the mouse sera. Furthermore, it has also a long duration, and there's still biological test variability, mainly because of the mouse phase in this test. So there was still need to move over to an in vitro potency test, which we started to develop around 2015, and during the development, we consulted the Paul Ehrlich Institute, the official medicine control lab in Germany, because they have the expertise on 3R testing, but also that ideally, if we would have a successful in vitro potency test, they might also consider it to introduce it for the EU release of our vaccine. Next slide, please. Maybe a little bit background also on what are the challenges in development and harmonization of an in vitro potency test for veterinary vaccines. Well, veterinary vaccines, they all use an adjuvant, human vaccines not. So that's a big difference. Another big difference is that human vaccines, the variability between the strains is less diverse as for veterinary vaccines. So with veterinary vaccines, you need to uncouple your G protein, your rabies G protein, from your adjuvant. So that might be adjuvant specific. We have a specific test for our aluminium phosphate, which might not work for aluminium hydroxide or another adjuvant. So it's really a specific test. We searched for it for quite some time. And another hurdle is that the rabies vaccine strains that differ between manufacturers. We tried in the VAC2VAC team also an antibody ELISA panel that was already approved by authorities, but it didn't work for our vaccines. So that's a pity, but that's mainly related to the fact that there are different strains used for veterinary vaccines. And then finally, what you also should consider is that if you use your antibody in your ELISA, that the antibody ideally should only bind to trimeric G protein, like Benjamin already mentioned, and ideally also it should bind your biological reference preparation, your reference vaccine standard that you use in your challenge test or use in your serology test. Okay, next slide, please. So our in vitro potency test is done with an alpha ELISA, which is comparable to a sandwich ELISA. We first set it up as an ELISA, and then we went over to the alpha ELISA. I will come back on the principle of the ELISA a little bit later on. But in essence, we just measure the G protein recovery in our vaccine. And because we already had the ELISA implemented as an in-process control test, we could use the same monoclonal antibody also for a finished product alpha ELISA testing. And we did it in 4 steps. The first step of validation was that we validated our pre-treatment method to uncouple the G protein from the adjuvant, and we had a very high recovery, so that was fine. The second step was, according to the pH 5 to 14, your antibody or your ELISA or alpha ELISA should quantify your antigen. It should detect the antigen integrity, let's say the 3D conformation, it should detect, and it should target an epitope relevant for protection. And the third step was just a bioanalytical validation according to VHGH guidelines 1 and 2. And the last one was the setting of the release specifications. And we did it obviously on the consistency and production approach. We analyzed a decade of batch data. And then the second step that we did, that we analyzed batches in our novel IVP test, which were already approved or which were already tested in the most challenge test. Next slide, please. Now, before we start, I think everybody and all manufacturers, they know that the antigen input correlates with your outcome in the mouse challenge test. However, because the mouse challenge test has such an enormous variability, it's often difficult to get a very high correlation. And I would certainly not recommend for other manufacturers to try to search for a correlation between your in vitro potency test and in vivo potency test. And I've seen examples in papers that manufacturers, they used 3,000 mice and finally got a correlation of 0.45, which is not a very high correlation. So it does not have a lot of added value, although people always ask for it. And it is a bit of a misconception that if you have a good correlation, that also that means that you have a very good in vitro potency test. The second thing that I already mentioned is that your monoclonal antibody or your antibody that you will use in your ELISA on the final product should detect the trimeric rabies G protein, because that is the immunogenic formation, and it should not detect the mono or dimeric glycoprotein, which is polyimmunogenic. Next slide, please. That the antibody, the MOAB that we use, can detect loss of integrity and loss of the trimer structure. That is shown in this in these two graphs. If you expose the vaccine to a heat or a chemical treatment, you will see that antigen integrity is lost. And this is also done to show that the ELISA test or the alpha ELISA test has just discriminative power. On the left side, you will see that the heat treatment, that already within half an hour or 1 hour, you lose approximately 80% of your ELISA signal, and this was a heat treatment at 50 degrees. And on the right side, you will see that if you use a chemical inactivant, and in this case it was beta-propylactone, BPL, the higher the concentration of BPL, the less ELISA signal you will pick up. So it will discriminate a loss in rabies protein content. Next slide, please. Then another requirement from the pH 5 to 14 is that ideally your test or your monoclonal antibody should target an epitope relevant for protection. We already knew from another group who characterized our MOAB that the MOAB was directed against the antigen 2 site on the rabies G protein. So we already knew that it has neutralizing capacity, but we always also tested if it has neutralizing capacity in a rapid fluorescence focus inhibiting test, and that is shown on the left side. It's probably too small to go into details on it, but the MOAB that we used has neutralizing capacity. And on the right side, you also see a similar graph that the MOAB is inhibited if you add the WHO International Standard Anti-Rabies Immunoglobulin in the assay or in the pre-treatment. So, and the rye that is used to standardize some of the rabies in vivo assays. So indeed, it has an epitope which is related to protection. The third step that we then did is that we did the alpha LISA test validation. And what is now an alpha LISA? An alpha LISA is very much like a sandwich ELISA, in which you have two antibodies which bind the antigen, and just in this case it will be the G protein, but the only difference is that the connected to the antibodies are two beads, and these beads, they can be excited by a laser beam, and if you see the alpha bead, alpha donor bead, if that is excited with a laser beam, it will produce immediately a high energy oxygen molecule, which then very quickly diffuses. And if there's a acceptor bead in the neighborhood, this acceptor bead will, because of the oxygen, will emit light. And this light then is detected with your alpha LISA detection device. And the amount of light that is emitted is proportional to the amount of antigen that is in your sample present. So the alpha LISA was validated according to the VHH principles for linearity, accuracy, precision, specificity, and robustness. And then the final step was setting the release specifications. It was again, like previously already mentioned, we used a consistency approach in production. We analyzed 10 years of historical batch challenge data. We had a very high potency on average, and the lowest vaccine that we released was, I think, around 3.6 international units. So we knew that we were already safe because we always formulate on a fixed amount of antigen. So that is confirmed then. The second step was that we analyzed batches that were already by the challenge test tested and approved, that we also tested them in the alpha LISA test. And then this outcome, it was, we tested, I think, 25 batches in the alpha LISA test. And we took the tolerance interval of these 25 batches and used that to set the threshold for release, for batch release. And the same threshold is also applied for the stability, for the ongoing stability and end of shelf life specification. In 2021, we wrapped all the data together and prepared a variation package in a work sharing procedure. And in 21, it was approved in the EU. Also outside EU, we have several countries who approved the package. And also in 22, we transferred the in vitro potency test to the Paul Ehrlich Institute in Germany. And we were very happy that they could rapidly also implement the test at their site. And that's still, I think, a very good collaboration between the two partners. In EU, we had approximately 29 countries who approved the test with the work sharing. As you can see, outside the European Committee, we have another 8 countries or 9 countries who approved it, and countries in Africa, Latin America, Asia, Middle East, and Australia have approved it. Most questions still focus on the correlation between in vivo and in vitro testing, and we get questions on how we set the release specifications and how an alpha ELISA, the principle, how it works. I think that's it. I would like to thank obviously AFSA, but also the 3R group from MSD, Fred Sulsov, who is the project lead, and Imke Cross, the project head, was the project head, and obviously also the VAC2VAC committee and the PAI for implementing the test at their site so rapidly. So thank you for your attention.
Thank you, Peter. We still have a following up question from the previous one. So it's about the use of BRP as a reference standard, as also MSD shift away from the BRP and adopted a similar approach as Merial and Boehringer Ingelheim.
Correct, yeah. Obviously, the BRP is used as a reference vaccine in the challenge test and in the mouse serology test. So we already had some kind of reference and we could calibrate it also in our in vitro potency test by looking at the challenge outcome. The thing with our MOAB was that it does not have a very high affinity for the BRP. So that's indeed a let's say, a slight hurdle to take. But you can do it even if your ELISA antibody does not cross-react 100% with your BRP. It's still possible to do it. And mainly you can use obviously historical data and consistency in production data.
Thank you, Peter. I think that we can move to our next case study.
Yeah,
thank
Ignacio's
you, Peter.
turn now.
Thank you,
okay, thank you.
Ignacio. Thank you.
Okay, perfect. So good afternoon, good morning, or good evening, everyone in different time zones. So I'm Ignacio Gisbert. I represented Zoetis for the variation for the replacement of the in vivo serology testing by an in vitro by ELISA. And then if you can go to the next slide. So indeed, we had a multivalent containing rabies vaccines and also monovalent rabies vaccines. So as we had some of them registered centralized in Europe, we manage all the products together centrally via the centralized procedure with PAI as the RMS. So the data package for submission was basing on the further description of the methodology and the reagents used. And then here it's important to emphasize that Zoetis' approach was to use a commercially available kit. Then we validated that method according to BICH guidelines 1 or 2, as my previous colleagues have mentioned. And then the big difference here with the previous presentations is that indeed Zoetis had used the BRP, so the biological reference preparation, to qualify an internal reference standard. So again, we will not use it as a relative unit, but rather as a reference standard for quantification of the glycoprotein content. Then as mentioned before, go back. So as mentioned before by Kat, we use consistency data from recently manufactured batches and stability studies under normal conditions and forced degradation conditions. If you can to the next slide. Yeah. So we have been talking, I think, the whole afternoon about the European Pharmacopoeia 5.2.14. So I will not emphasize more. I think that's a take-home message for everyone here. If you want to replace an in vivo method by an in vitro, please go there, look at it. There is a lot of valuable information, and hopefully that will be also incorporated on the new VICH guideline mentioned by Laura before on previous presentations. Then with regards to the data package, we have the consistency data. That's for the multivalent. So you can see that we have the GP content, which is stable and consistent during manufacturing. And then we have below with no resolution of power with pass or not pass the in vivo method. And then we have the same type of information available for the monovalent vaccines. So next slide. So as my colleagues discussed, one of the key points of the variation was how to set the specification. So we started with the maximum content, and for this, as Kat has mentioned before, consistency, consistency, consistency. So you have a lot of historical data, you have to mine it and then arrive to a consistency approach for your specifications. In our case, a specific case, we were also able to mine some data from previous laboratory safety studies in which we had a known concentration of glycoprotein, which indeed help a lot to set the specifications, the maximum specification. Then with regards to the minimum content or the minimum specification, we have first an end of shelf life. So that will be the minimum content of glycoprotein in order for the vaccines to be effective. And that we were able to mine that data also from a known concentration of glycoprotein in an efficacy study. Again, those studies were not done on purpose for this variation, but rather we mined the data from already pre-existing laboratory or field studies. Then once we have the minimum efficacious dose, what we wanted to have is how do we set the specs for the release. So for this, we base again on consistency data and also stability data and all the data that has been generated during the many years of the product on the market. So if we can move to the next slide. In addition to this, so we presented the stability data, which had a drop up to 40% of the glycoprotein content. And then we designed some force degradation studies, which are in line with the ones presented by my colleagues previously. So we did the heat treatment at 95, 65, and 42 degrees. And then we compare both serology and the new in vitro method. So as you can see, there is 100% correlation between both methods, with the difference that the new ELISA at this quantitative has a high resolution power and can indicate better and can pick up a degradation of the antigen in an easier way. So next slide, please. So then we wanted to emphasize on the procedure. So here, when we started the procedure, the main question we received is indeed if the assay was suited for purpose or not. And then that comes from the fact that in Zoetis, we use the approach of use a commercial kit. So the authorities were asking us, what is the monoclonal antibodies used in this kit? Is it capturing the relevant conformation and the relevant structure of the glycoprotein? So indeed, we provide all the information that was available by the kit manufacturer. We confirmed that it was indeed a neutralizing antibody, but however, the monoclonal antibody was able to bind both the monomeric and the trimeric form of glycoprotein. So then the authorities wanted a follow-up question on which one they wanted to make sure that the monoclonal antibody was indeed detecting only the relevant conformation of the glycoprotein. So the approach of Zoetis to respond to these questions was literature, and then practical demonstration of the ability of the monoclonal antibody to detect the right conformation. So if you can move to the next slide. And the next slide. So just for those of you that are not that close to the rabies virus, so it's a single-stranded negative sense RNA genome that it contains structural proteins. From those, the relevant here that we are speaking all the time, it's glycoprotein that you can find it on the right corner on the illustration. And then if you move to the next slide. So the glycoprotein for rabies is mediating both the viral recognition and the attachment of the cellular receptors. And it's also the major target for neutralizing antibodies, as explained before. Then in the viral surface of the virus, it's structurally present in an heterogeneous form. So that means only a portion of it will be on the trimeric form, but the other one, it can be found in dimeric and monomeric. However, and that was also explained by my colleague, only there is a current equilibrium between the pre-fusion conformation and the post-fusion conformation, and it's pH dependent. And again, this equilibrium is reversible. So depending on the pH that you encounter your antigen, you will find your antigen in one of the conformations or the other. Next slide. So in order then to demonstrate practically what we were saying about what we found on the literature, then we did a buffer exchange of our antigen, and we changed to different buffers, lowering with the pH. The expectation here is to show on the ELISA that the ELISA results are higher with a pH around 7, and then as long as you decrease the pH, then it goes to decreasing the signal for your ELISA. And if you go to the next slide, you can see the actual results. So we had a vaccine that it was not for release, but the rather theoretical GP content with a high content of glycoprotein, in which we leave it untreated, and then we did a buffer exchange with the different buffers show on the different pH. So as you can see perfectly on the image, once you go below pH 6.8, it's the ELISA is no longer able to detect with the same ability the glycoprotein content. And then in order to make sure that we were able to present the data for the procedure, as we were a bit concerned with the coating buffer, and we know the rabies glycoprotein is on an equilibrium that is reversible, maybe the coating buffer will affect the conformation of the glycoprotein. We decided to design a quick and easy experiment, which is the dot plot, which is basically an ELISA, but without the buffers for coating. So you have a nitrocellulose membrane, you put your antigen, you dry, you block, and then you add your primary antibody and secondary antibody for repeating. And then if you go to the next slide, I think it's also very graphical. You can see as long as you decrease the pH, the concentration is getting lower. And again, you still see a dot on a low pH because it's still an equilibrium. So even though the majority of your glycoprotein is on the pre-fusion conformation or post-fusion conformation, you still have some of them that are still on the other conformation. So the lessons learned we kept for the procedure is the importance of the force degradation studies and the importance to select the right form and the right conditions. So in this case, so we started with only temperature, and then we realized that pH would have been considered very important, and indeed was at the end the key point that granted the approval. Then as the message that I think we are all taking home, the importance of consistency data, and then where possible with very old products is not always the case, efficacy and safety studies always facilitate the procedures. Then, as my colleagues say, we have approvals not only EU, but also in other countries, for instance, Thailand. So as Benjamin was speaking, there is the possibility, and so it is to succeed to register the new in vitro. Then the manufacturing partner with Zoetis, it's also been able to register it in other geographical areas such as Ukraine, Turkey, and Bosnia. And I think with that, I will say thank you, and I will open it for questions, if any.
Yeah, Laura, if we see if anybody's got any questions for Ignacio, and then we can go to my last wrap-up ones.
At this point in time, there is none. It's just a general question about the possibility to release the protocol.
Yes, it seems like a few people are very interested in us sharing,
exactly.
Yes, sharing a method for the desorption steps. So I think we can probably, I'm not sure if there's publications on desorption steps. I'm not sure if we're all using the same method. It's probably all slightly different. But we can take that one offline. We can see what approach each of us are using, and potentially as a follow-up to this, share some references and some basic methods for the desorption step. Clearly, if that's something that's a barrier for some of those international companies in developing these tests, there's value in us doing that. So we can do that for everybody.
Absolutely.
Maybe a small comment from MSD Animal Health. We are considering to write a paper on the whole method, and as soon as that is published, it will become available.
Yeah, and there is already a publication out there on the BI method that Benjamin provided. So we can make sure that there is some information that we can provide on both any publications related to the methods and desorption approaches. Desorption steps are necessary for a number of vaccines that contain adjuvants. So it is something, I think it was even talked about with Invactivac. I need to remember, Carmen, but we can go back and see what we've got that would be useful to share with everybody. Yeah, so just to wrap up, thank you to Ignacio, Peter, and Benjamin. So as you can see, a lot of common themes, but 3 examples, different methods, but the same sort of approach. And as Jean-Michel said in the chats, in human health, they're looking at a single common method, but there are some differences. Obviously, all the current veterinary vaccines are adjuvanted, so we're dealing with a slightly different approach, although potentially those antibody sets in human health would work for these. But what's critical, and you see this right across the board, is the ability to demonstrate the suitability of the method to detect an immunologically relevant antigen. Establishing the discriminatory power, so ensuring the method could detect both subpotent and substandard batches. So you saw that in the data from everybody here that so Peter showed you that the method, there isn't really a correlation over the board between the in vivo and in vitro methods, but as Ignacio showed you, certainly with the Zoetis data, the pass-fail, there was a pass-fail correlation, e.g. once you'd established your specifications for the in vitro method, you could ensure that if it failed in the in vitro, it would have failed in the in vivo and vice versa. So there was certainly a relationship, although not a full correlation between the methods. And as Peter mentioned, that's because the ability to really validate the in vivo tests would be very, very difficult right now. And also the criticality of using the consistency approach to establish relevant release and end of shelf life specifications. So you saw from all of these that we were establishing specifications not just for release, but also end of shelf life. So ensuring that we understood how much loss there may have been over shelf life, and ensuring that those release specifications were set to ensure that you always had a minimum level of efficacy. There's one more slide, I think, Laura. Yeah, and these are just some key messages from the rabies approach. So as we've touched on, the single method may not be suitable for all products, and that is the case here with the rabies example, but it may also be the true for other antigens. The BI method, we tested all manufacturers in Vactivac and it didn't work for everybody. That there is because one of the antibodies is highly specific for the Pasteur strain in the BI vaccine. But as Jean-Michel mentioned in the chats, that there are potentially antibody sets that would work for everything. Using the same approach in all cases increases confidence in both that approach and the outcome. So while we're not using all the same method or the same antibodies, the approach and to the validation, to demonstrating the discriminatory power was very, very similar across the board. We've touched on that about the direct correlation, but they are, like I say, they are consistent in their ability to ensure potency. The specifications for existing products are based on historical consistency data. In some cases, with the last one with Zoetis, there was some challenge information where we did have theoretical glycoprotein content, and that was based on the fact that we knew what the antigen content was of the antigen going into it. It wasn't that we tested the finished product, so it was a theoretical content. But what's important is that there has not been any need to repeat challenge tests to establish specifications, nor any need to run challenge tests to establish a reference. As we've talked about in all cases here, we were either using the BRP reference simply as a reference standard, or specifications were established purely on consistency based on the outcome of the method. And using the same method and stability data to establish both the release and end of shelf life specifications. So testing through shelf life to ensure those specifications. And as you saw, all three methods have been accepted countries around the globe. Thailand, an interesting one where I said we'd presented these in historical order. So interestingly, you would see that BI did not get approval in Thailand, although down the line, both Zoetis and MSD did. So clearly there's been some change in approach there. And in all cases, it does take a significant amount of time and resource to develop the method and generate the data, and then to establish and support the specifications and the regulatory approval. So this is not something that's quick. It's not easy. It takes a lot of time and investment, but it does give us better product consistency. It was touched on the release testing time is significantly reduced. So it's like 1 to 2 days to do an ELISA versus 3 months for a challenge potency test, up to. It reduces our costs and it enhances supply continuity. I think Peter or Benjamin touched on the fact that many of these rabies vaccines are used for vaccine banks towards trying to eradicate rabies. So again, it means that we have, because the testing's quicker, products go out with better shelf life on them. So that's important for supply continuity and the speed with which we can react and supply more vaccines. We don't have any issues with the animal supply for the potency tests. That is a continuous issue. And we have less no tests or test failures. So we've got less repeat testing. And you can imagine having to repeat an animal test versus repeating an ELISA is a world away. So time is very important. And just a final comment. In the rabies case, the relevant epitope was well established and well defined, but that's not always the case. So we wouldn't want anybody to think that this nice rabies example is going to be possible in every single case. And that really is what the core of Pharmacopoeia monograph 5214 and the future VICH monograph will be about, that you need to take each of these case by case. But the general approach will be the same in every single situation. And I think, Laura, that's the end of it. And I see one question in the chat.
Yes, we have one question about the acceptance of these different methodologies in the US context that historically has been more in favor of a unique approach, and then a comment from Jean-Michel about the correlator protection and how it has been established in the human case with the screening.
Yes, so let's take Mark's question first. So
yeah,
yes, the elephant in the room in all of this with rabies is the USDA and the US approach. So I think most people online know that the USDA would prefer a single method. Because they do all their own release testing, they would prefer to have a single method. And as of right now, we believe there would be an expectation to use a more relative potency approach where companies would need to repeat challenge work to establish and reference, and that's been historically the approach. We hope to go back and discuss this further with the USDA again. We will certainly, I think we've got Amy Gill online from the USDA, and we'll certainly share this presentation back with Geetha Srinivas and the USDA folks and with AHI and continue the conversation. I think it's going to be a longer haul, but it's useful in all cases to share the success we've had in Europe and globally with this, which will hopefully move things in the right direction. But yes, the US is still a challenge. It's not a challenge we figured out how to overcome yet, but we continue to work on it. And this was a nice opportunity to pull together the different experiences with Europe and to be able to see across the board how we've managed to have success globally with this outside of the US. So ongoing discussion with regards to the US, unfortunately. Jean-Michel,
yeah,
it's a work in progress. Can Jean-Michel speak himself, Laura? Can we unmute Jean-Michel? Oh, I can't hear Laura now. I think you've muted yourself, Laura.
Yeah, I just allowed Jean-Michel to jump in. So
okay, Jean-Michel, please, it's easier for you to speak.
Yeah, thank you. Well, I was wondering if there is a possibility to compare your method with the human methods, because I think that actually the FDA will probably accept the method for human vaccine. I say probably because it's not really true for the moment, but in the next future it would be accepted, I think. So my question is, could it be possible to compare your method with the human ELISA? I know that there is adjuvant, but we don't know if it's working with adjuvant and without adjuvants.
I think we could certainly do that, Jean-Michel. I mean, certainly each company could, once you have that, once you complete the BSP project and that method is finalized, I think we could certainly look and see whether it works. Most of the desorption methods are relatively straightforward. It's kind of incubating on a plate overnight. So while we might need some modification of the ELISA method, we could certainly check whether the antibodies themselves would work. We could have a go at that. That's certainly something we could do. And if the FDA, let's assume the FDA finally accept it for human vaccines, then it's something that we would hopefully also be something that might support further conversation with the USDA on how we approach this in the US. We know certainly that for many international markets, if the vaccines are being produced and supplied out of the US, we will continue, even if an alternative method was accepted in a local market, the batches will continue to have to be tested by the USDA for release because it's based on the outline of production. So the next kind of major hurdle for this globally for us in the veterinary sector is certainly working with the USDA to find a path forward, and certainly potentially looking to see whether the human health method works on the veterinary vaccines is something that's worth doing. It's a different story as to whether companies would want to move to that method. I think that would need to be an individual conversation.
I understand your position, but my fear is that USDA can be, you have to be very careful, because you can have the NIH and ELISA in parallel for your release. So in the future, it could be helpful to convince USDA by the data in comparison with the human ELISA.
Yes, no, it certainly may be useful for us to do this just in the overall trying to get approval of that type of method. So it's certainly worth exploring. And we'll be very interested to see, fingers crossed, when the FDA when they get the first FDA approval for it.
Sure, of course, yes. BSB 148 is going very well.
Yes, yeah,
we
and
are
I
in the
think
middle of the phase 3, and we are still expecting some result, but we are actually, there is no issue, main issue. We'll see in the future.
yes, absolutely.
Thank you very much, Kat.
Thank you, Jean-Michel. Laura, does anybody else have any questions? I think we've answered all the questions in the chat.
I think so.
And we've got a follow-up action to share something about desorption methods.
Exactly.
And
exactly.
we can do that.
Yeah, we will follow up, and we will try to share materials and information through the AFSA website, so we can actually upload the materials there. And unless you have any final remarks, Caps, in the interest of time, I would move to our next session.
No, I don't, if there's no more questions, Laura, but if anyone does have questions, feel free to put them in the Q&A, and I will try and answer them while we carry on. Oh, no, as I speak.
Thank you.
Thank you, Amy.
You,
Amy, I will send the presentation to both yourself and Geetha. I did chat with Geetha about this a couple of weeks ago, so she knew what we've been doing with this.
Thank you, Kat, but also thank you, Benjamin, thank you, Peter, and thank you, Ignacio, and also thank you to Jean-Michel for jumping in on this important topic. So we are actually moving to the Clostridial case study. So I'm actually happy to move to our next speaker, that is Laura Astle from HRA UK, that is actually going to present about the work that MHRA has done on the tetanus vaccine in vitro potency assay. Laura, the floor is yours. You can share your screen.
Thank you. Let me just, hopefully this will work.
Yeah, it's coming up. All good. Thank you.
Yeah, all good. Thank you. Yeah, so I work at the Medicines and Healthcare Products Regulatory Agency, previously known as NIBSC, and we were a partner of the back-to-back consortium working on development of in vitro assays for human DTP and veterinary tetanus vaccines. So the presentation today is on the development of an immunoassay for potency testing of tetanus vaccines, and it's going to be a focus on the veterinary results that we obtained. And many of the messages from the rabies case studies are echoed here, as you'll see. So regulatory authorities require a potency assay or a potency indicating assay to be performed on every lot of vaccine that's released. Potency is a critical quality attribute and is a test of the functional integrity of the antigen, i.e. it determines whether the vaccine can do its job properly and stimulate the desired immune response in the target species. And it's also an important indicator of stability. Potency also provides important evidence and assurance for consistency of production, i.e. that new vaccine lots are comparable to those originally used in clinical studies for which efficacy was demonstrated. So currently, as we know, potency testing of tetanus vaccines relies on the use of in vivo models. Traditionally, potency is determined using a challenge test, which results in severe suffering for the non-vaccinated or only partly protected animals. And efforts have been made to replace challenge assays with serology assays, which represents a refinement of procedures since the requirement for toxin challenge is removed, and also we were trying to implement reduction schemes to reduce the total number of animals used. Nevertheless, these animal models, both challenge and serology, have significant limitations, including the ethical concerns, as I've just mentioned, the high costs and prolonged testing period, as well as high variability and poor discriminative power, raising questions about their relevance. So the Vac2Vac consortium was initiated with the aim of developing new tests that support the use of the consistency approach in batch release testing of established vaccines. And for tetanus, the quantity and quality of antigen present are key factors to be monitored as part of the consistency testing. And we've developed an ELISA that's intended to provide a quantitative estimate in relative terms of the tetanus antigen content. The ELISA uses well-characterized monoclonal antibodies directed against functionally relevant epitopes on the toxoid that are sensitive to changes in the quality of the antigen, so it covers both these factors to be monitored. The full characterization of the monoclonal antibody reagents has been published, including binding to native, detoxified and adsorbed antigen, binding to heat-altered antigen, as well as affinity and epitope competition studies used to identify a suitable pair for a sandwich ELISA. So this schematic just shows the steps in the ELISA, so coating with the captured monoclonal antibody, blocking an addition of vaccine samples, followed by detection of the bound antigen with the detecting monoclonal antibody, conjugate antibody and substrate. The assay performance characteristics of the ELISA have been evaluated, and the assay has been shown to have many advantages over the in vivo potency tests. The work on the development of the ELISA assay has also just very recently been accepted for publication. And this table just summarises the characteristics of the ELISA compared to the animal potency tests. You can see that the time required to perform the assay is significantly shorter, 2 days in comparison to 4 to 6 weeks. No animals are required. It has much higher precision than the in vivo assay and lower variability, and therefore much better discriminative power. So how would we see this assay being used? So across the middle of this slide is the different testing stages of a product throughout its life cycle, starting with early R&D and finishing with post-market surveillance. So in vivo assays still have an important role to play in early R&D and non-clinical development of vaccine products, as they provide a biological response to the candidate vaccine that can be readily measured in the absence of a thorough characterization and knowledge of the vaccine's critical quality attributes. However, for new vaccines, in vitro assays can be developed at the same time as the in vivo assay, so that they can replace the in vivo assay during development and at the time of licensure. For the legacy vaccines, as I've just mentioned, the in vivo potency assays remain part of the routine QC and lot release testing. And the aim is to replace these in vivo potency tests with the antigen ELISA, either alone or in combination with other in vitro assays as part of the consistency approach. So in the Vac2Vac project, a wide range of samples were obtained from 2 human vaccine manufacturers and 4 veterinary manufacturers. This slide shows the dose response curves for samples from the 4 veterinary manufacturers, and they include both upstream toxoid samples and final vaccine products. The vaccine products contained a range of aluminium and non-aluminium adjuvants, as I've listed here. And I've also made a note to say that all the vaccine samples shown here are diluted whole vaccine, i.e. they're diluted in the presence of their adjuvant. As you can see, suitable dose response curves were obtained for all samples except 1 trivalent product from veterinary manufacturer A. And this particular product contained an aluminium adjuvant, and the antigen detection was significantly improved when the sample was desorbed. Overall, the magnitude of the response correlated well with the type of sample titrated, with the highest response seen for the toxoid samples compared to the final vaccine products. So we assessed the ELISA specificity by testing dropout samples for a range of vaccine products that contained all components of the vaccine except the tetanus toxoid. And no response was observed for any of the dropout samples except the one containing a Hib component conjugated to tetanus toxoid as a carrier protein. This isn't surprising, and we have previously shown that tetanus toxoid conjugated to polysaccharide could be detected in a similar sandwich ELISA assay, but the amount detected was considerably lower than the conventional unconjugated toxoid. The intermediate precision of the ELISA method was determined by testing a human and a veterinary vaccine product one reference batch and one test batch for each across duplicate plates on several days, and was found to be very low in the range of 5 to 6%. To assess the dilutional linearity of the ELISA, one of the dropout vaccines was spiked with increasing amounts of tetanus toxoid, ranging from 25 to 400% of the normal vaccine dose. As you can see here, the target and observed results have good agreement. The fittest slope was not significantly different to 1 and an intercept not significantly different to 0. So this data also shows that the ELISA can detect 25% changes in the antigen content, something that would be difficult to see using an animal assay. So as I mentioned at the beginning, the potency assay has traditionally been used as a measure of stability to detect changes over time in the potency of a product. So in the Vac2Vac project, we tested the ability of the ELISA to detect quality changes in tetanus toxoid for a selection of aluminium adjuvanted and non-aluminium adjuvanted vaccines. The conditions that we used to produce these water samples were heat treatment, oxidative stress with hydrogen peroxide, and shear stress simulated by sonication. For the heat and oxidative stress studies, I've just shown some representative examples of the veterinary samples tested. So in the first graph, vaccine samples were incubated for 8 weeks at elevated temperatures of 37 and 45. And the antigen content of these samples was measured and compared relative to the sample stored at the normal storage temperature of 4 degrees. And the graph shows 3 different non-aluminium adjuvanted products, 2 monovalent vaccines and 1 monovalent vaccine, all of which showed a loss in antigen content to varying degrees following storage at the elevated temperatures. And the same was also observed for the aluminium products tested. Similarly, the middle graph shows a non-aluminium adjuvanted vaccine product incubated with increasing concentrations of hydrogen peroxide for 1 week, and there was a reduction in the amount of antigen detected with the high concentrations of hydrogen peroxide. So the last graph shows a tetanus toxoid sample diluted to final vaccine concentrations and subjected to an increasing number of sonication cycles, and there was a clear decrease in antigenicity with an increased number of cycles. And this correlated well with an increase in aggregates that was observed by HPLC and performed by another partner in the consortium. So in some cases, desorption may be required to allow for suitable detection of the antigen in the ELISA, since desorption onto an aluminium adjuvant can mask the epitopes on the toxoid that the monoclonal antibodies are directed against, preventing or reducing binding. As I mentioned previously, antigen detection for a veterinary trivalent vaccine from manufacturer A was significantly improved when desorbed compared to the whole vaccine. So to further investigate how much of the antigen is accessible when adsorbed onto an aluminium adjuvant, we performed desorption studies and compared the amount of tetanus toxoid detected after a desorption step to that detected in the whole vaccine sample. And we found that for the veterinary products containing an aluminium hydroxide or aluminium potassium sulfate adjuvant, the proportion of antigen detected in the whole vaccine sample was approximately 40% compared to after a desorption step, whereas most or all of the antigen was detected in the whole vaccine samples containing an aluminium phosphate adjuvant. So with these studies, it's important to remember that the desorption step may not be 100% efficient, and the effect of the desorption process on the antigen is not fully understood. In terms of monitoring consistency of production, however, it may not be necessary for 100% of the antigen to be detected. So in-house qualification assays confirm that the ELISA has very good reproducibility, and to further investigate the suitability of the ELISA for monitoring batch-to-batch consistency, multiple lots of the same product covering a range of different ages were tested, where age refers to the time from the date of manufacture of the final bulk to the date of testing. So this graph shows the results for one of the 3 products tested, with batch ages ranging from 1 to several years. The between batch variability observed was only slightly higher than the within batch variability observed in the qualification studies, demonstrating that the assay should work well with real-world samples and will be suitably powerful for detecting quality differences between different vaccine batches. And there was also no evidence of any toxoid age-dependent impact on estimates of relative antigenicity. So a move away from in vivo potency assays to the use of in vitro assays as part of a consistency approach will almost certainly require development of product-specific acceptance criteria as opposed to expression of potency in international units. And to this end, we evaluated the suitability of different materials to act as a reference preparation in the ELISA by examining the similarity of the dose-response curve shapes. The reference materials evaluated included the WHO International Standard for Tetanus Toxoid 16302, the International Standard for Tetanus Vaccine Adsorbed 08218, manufacturer-matched drug substances, as well as a range of manufacturer-matched drug products. The sample and reference curves were considered to be similar if the difference between the upper or lower asymptotes fell within minus 0.05 and plus 0.05, and if the ratio of the slopes was between 0.9 and 1.11. So here are some examples of the results we saw. So example 1 shows curves which have the same profile, i.e. no differences observed in slope or asymptotes, and for which we would consider the reference suitable to use to calculate a relative antigen estimate for the test sample. Examples 2 and 3 show curves where we saw differences in either the upper asymptotes or the slope ratio between the reference and test sample, and where we would conclude that the reference is not suitable. But you can see the sort of differences we observed are very small in all cases. So overall, small differences were observed in the upper asymptotes and slopes for some of the products compared to WHO standards, but generally not against the drug substances from the same manufacturer. And the results suggest that for many products, there will be different options available for the choice of reference standard to use in the assay. So finally, we've also demonstrated successful transfer of the method to other laboratories. Studies were carried out between us and 4 industry partners across 5 different sites. The protocols, including the plate layouts and critical reagents, were provided by MHRA, and the same vaccine samples were then tested at both sites. So at least one representative vaccine product was included in each transfer study, and 2 products were included in 3 of the studies. And for each product, 2 batches were tested, one assigned as a reference and the other as a test sample, so that relative antigen estimates could be calculated. Each lab performed 3 independent assays with 2 plates per assay, and all the raw data was returned to us for analysis and comparison. So we calculated the intermediate assay precision, and as you can see, this was low, less than 15% for all the products tested. And the GCV obtained by the partner lab was within 5% of that obtained by MHRA in all cases. In addition, the relative antigen estimates obtained by the partner lab and MHRA were within 10% of each other for all products except this one highlighted. So in conclusion, proof of concept has been demonstrated for the tetanus ELISA, including evidence that the assay may be stability indicating. It's been shown to be suitable for testing a wide range of human and veterinary tetanus vaccines. Assays are robust and successful transfer to other labs has been achieved. And vaccine manufacturers in the VacToVac consortium are further exploring the use of a monoclonal antibody immunoassay approach as part of as a potential substitute for current in vivo potency assays. The purified monoclonal antibodies used in this ELISA are available from the NIVS catalogue for anyone who wants to establish and validate these methods, removing one of the most common barriers to development of the alternative methods, which is availability of the critical reagents. So finally, just to acknowledge colleagues from MHRA, Intravac and Sciensano who contributed to this work, and in addition to all the industry partners in the VacToVac consortium, the project coordinator and other consortium members. Thank you very much.
Thank you, Laura. We have a few questions on the Q&A. If you can quickly go back to the suitability of the methodology for multivalent complex vaccines. For example, in the livestock products, it's not uncommon that there are multiple Clostridium species in addition to bacterians, viruses, and adjuvants. So if you can comment on that. Thank you.
Just going to read that question again because I didn't catch it fully.
Of course.
Okay, so is it asking about the specificity, I think, for other Clostridial species?
Yes, if
which
it's,
we have,
if it's,
yes, which
if
we
it's
tested
suitable.
for in the specificity where we tested dropout vaccines, and that did include dropout veterinary vaccines, which had other Clostridial species present, and there was no signal from those.
Thank you. And then the other question is about whether you have compared the assay with the radial immunodiffusion.
No, we haven't done that, no.
And if there are any type of controls on the ELISA? That's the last question. Thank you.
I'm trying to think what we had. So we had, so when in the Vac2Vac, we included WHO standard either as a reference or as a control, just to show that the assay was working. Routinely, if you have a product-specific reference, I don't think you would necessarily need to have an internal control, but yeah, we haven't gone as far as to define what should be included as the controls.
Okay. Do you have any more comments, Laura?
No.
Okay. So thank you very much. If you have any other questions to Laura and the work that has been done within MHRA with ELISA, please just add them into the Q&A. I would like to now welcome Anne Thomas from Zoetis, that is actually going to give us an overview about the work that's industry has done in the implementation of the Clostridium tetani ELISA. So Anne, the floor is yours.
So thanks a lot for your introduction and to give us the opportunity to share our feedback on the applicability of such ELISA in vet industry. So I'm Anne Thomas, working in biological analytical sciences at Zoetis Belgium, and I was part, as Laura and Paul, of the Vac2Vac consortium. So the difficulty for the vet industry is that such tests should be done in GMP environments, so in QC labs in different countries to release each commercial batch. So that's why there are different critical steps, parameters that are listed on the left and that we will analyze in more details in the next slides, but for which the Vac2Vac consortium and the work of Laura and Paul as a help to already overcome some of these steps. Next slide, please. So the first step, as you have heard already in different presentations for such in vitro ELISA development, is to identify the reagent and so the antibodies. And so, as presented by Laura in a presentation, there were multiple antibodies that were evaluated during the Vac2Vac consortium. So deeply characterized, I list several examples of tests. Also, the quality of reagent was assessed by different tests. And so it came out different antibodies that were the most appropriate for the development of tetani ELISA. The second point that was mentioned in Vac2Vac is to ensure that this reagent would be supplied during many years, because it's what we have to do as vet industry, is to release batches during decades. And so Laura has shown there is a central production, purification of the antibodies and a global distribution. So through a catalog and a master material transfer agreement to fill, and then anybody can order this reagent, as it's the aim that these reagents are available. Next slide, please. A second critical step that was done in Vac2Vac was to develop the assay that quantify a relevant epitope, and so Laura has shown the different data. Multiple products were tested. Also, altered product was tested. We have even changed slightly some conditions like incubation and so on, and the test was robust. So it's really something really helpful for us. Now, we need to, for each product, we need to optimize the test because we have discussed each product contains different components. The matrix is different, so we might, for some case, do an extraction of the antigen, for some other product not. So that's why now for each product type, we need to have an optimization of the DSA. We might have also some constraint due to the country, depending on software to use. We need also to adapt the reference to use, as we have also discussed just previously. So that's the next step for the company to then optimize DSA for each final product matrix. And once it's done, next slide, please, to validate. You have seen, Nora has shown that already from VAC2VAC work, there were promising results regarding specificity, linearity, accuracy, and precision. But the different companies have to validate for each product, so following the ICH guideline, and you see the different parameters for validation. Another critical step that we have already discussed earlier is the setup of specification, because then we need to set up specification for each product, and it takes a lot of time, as mentioned by Kat, as we have to compile data, historical data for each product type. And for tetany, the link to the flocculation test, DLF, might be difficult to establish. So it's something that might be complicated to get. So it's also the setup of specification, a long step after validation. Next slide, please. And then last but not least is that, as I said, this test should be run in routine for release and also stability testing in multiple labs, multiple operators during decades, and in a GMP environment. So then we have to transfer this test, which should be robust in different labs. Now, during VAC2VAC, there was an interlab study that was run where we had already promising results of reproducibility, but it's still a long way to do for each product. Next slide, please. So in conclusion, the vet industries are working towards company-specific ELISA approach to substitute the current in vivo tetany potency test. As we said, each vaccine matrix needs specific assessment, for example, for antigen extraction, yes or no, for the validation and the setup of specification. But having reliable reagents, as we have mentioned, for these antibodies and already a robust antigen ELISA helps to move forward to have a tetany in vitro potency test for final product. Thank you.
Thank you, and we have a question on the chat. It's about again sharing the protocols about the absorption of the aluminium hydroxide. So I wonder if again between Laura and the experience from industry, you have again specific protocols as it was in the case of the adjuvants in the rabies vaccine.
So we can look with Laura what was done in the interlab study. I know that for some we did not, and now it's to see what was done for the ones for which we had extraction. So Laura, do you remember which protocol you applied for when you had to extract?
In VAC2VAC, but we have also used sodium citrate in the past.
Did you hear?
Sorry, I took my headphones off. Can you hear me now?
Yes, we can.
Yes, so in the VAC2VAC, we used EDTA sodium phosphate for desorption, incubating overnight at 37 with the sample. But I think we also looked at 20% sodium citrate, but that didn't always work as well for all the products. But yes, we can share methods.
Thank you.
Thank you.
So there are no further questions at the moment, so but please keep an eye on the chat in case others are coming up. And with this, I would like to move to our next speakers, which is Eike Behrensdorf-Nicole from the Paul-Ehrlich Institute, that is going to give you an overview about the Pinnacle assay to determine the activities of the tetanus neurotoxin. So, Eike, the floor is yours. You can bring up your presentation.
Oh, thank you.
Okay,
Okay,
all up.
can you see my
Yes.
presentation now?
Perfectly, thank you.
Okay, that's great. So, first of all, I'd like to thank the organizers for inviting me to this webinar. And so, just a moment. So this is my disclaimer. And so this is an overview of the Paul-Ehrlich Institute. It's a German medicines authority and it's located close to Frankfurt. And it fulfills a variety of regulatory tasks as summarized here in this slide. And besides, we also conduct research and method development. And all these activities are focused on the quality, safety, efficacy, and availability of vaccines in the human as well as in the veterinary field and of biomedicines. So I'll start with an overview of tetanus toxin and why we are interested in this toxin. And after that, I'll introduce the Pinnacle assay that was developed in our lab. So tetanus toxin is produced by the bacterium Clostridium tetani, and it targets inhibitory interneurons and thereby induces severe muscle spasms. And in humans and various animals, especially horses, which are very highly sensitive to the toxin, this toxin can lead to death by breathing problems. And tetanus toxin is extremely potent. So about 1 to 10 nanogram of the toxin per kilogram of body weight can already lead to death in humans and also in many animal species. The lethal dose is in a similar range. And tetanus toxin that has been inactivated by a formaldehyde treatment is used as tetanus vaccine. And it's very important that each of the inactivated toxoid bulks must be tested for the absence of active residual tetanus toxin. And due to the high toxicity of the toxin, it's really important that reliable methods are used for the toxin detection to avoid any unwanted side effects. And for all tetanus vaccines for human or veterinary use, the European Pharmacopoeia prescribes that the inactivation must be tested by injecting the toxoid into guinea pigs. And the animals are then observed for 3 weeks. And if none of the animals shows any tetanus symptoms, the toxoid is then regarded as safe. And so far, there is no generally accepted alternative method. And as already addressed by the previous speakers, it should be kept in mind that animal tests have various disadvantages. So first of all, there are ethical concerns because the tests can cause severe distress for the animals. Then animals tests are often associated with a high variability and low precision, as they are influenced by many environmental factors or the health status of the animals. And besides, it's known that the guinea pig test which was introduced many decades ago, has never been really standardized and validated according to the modern standards. So for example, for the guinea pig test, even the detection limit is not known. Then the animal test has a long duration. It has an observation phase of 3 weeks. And animal tests are generally expensive because you need all these animal facilities. So At the moment, the testing of the safety testing of the toxoids still heavily relies on animal tests, but replacement by an in vitro method would be really preferable. And so, as our goal was to provide an in vitro method for these activity measurements, we had a closer look at how the toxin works. So each tetanus toxin molecule consists of 2 protein chains, which are linked by a disulfide bond. I'm not sure if you can see my mouse here. And so when the toxin enters the body, the heavy chain, this one, first binds to receptors on neurons via its binding domain in the heavy chain. Then in the next chain, in the next step, the translocation domain, which is also in the heavy chain, then inserts into the cell membrane and forms a transmembrane channel. So then, to this channel, the light chain is then transferred into the neuron. And at the same time, both chains are divided by a cleavage of this disulfide bond. And this cleavage then leads to an activation of the protease domain in the light chain. And the active light chain can then cleave synaptobrevin, a protein which is present on the surface of synaptic vesicles. And in healthy neurons, this synaptobrevin has the function to interact with some proteins on the inner surface of the cell membrane, such as SNAP25 and syntaxin. And this interaction usually is required for the release of neurotransmitters from the synaptic vesicles. However, if the synaptobrevin is cleaved by toxin-induced cleavage, then the neurotransmitter release is blocked. And this inhibition then results in the severe spasms that are characteristic for tetanus. So when we started to develop an in vitro method for tetanus toxin activity testing, we first checked if it is sufficient to look at only one single characteristic feature for detecting the active toxin. So we tested if a single receptor binding assay or an endopeptidase assay would be enough. So the results of an receptor binding assay are shown here in the upper graph, and the results of a endopeptidase assay are shown in the lower graph. And you can see that in both cases, all toxoids that we tested showed high signals already when tested at low concentrations. Although all these toxoids that we used had previously passed the animal test. So no actual toxicity was present in them. So this means that in these single tests, the signals do not reliably correspond to in vivo toxicity, and no reliable discrimination between active toxin and inactivated toxoid is possible with such single assays. So then, to mimic the relevant steps of toxicity more closely, we then combined a binding and an endopeptidase step for specifically detecting active toxin based on its receptor binding and proteolytic characteristics. And this method is called binding and cleavage or binnacle assay. So in the first step of this assay, the toxin binds to receptor coated microplates. And in the second step, we then add a reducing agent, which then leads to the detachment of the light chains from the heavy chains. And the supernatant with the free light chains can then be transferred to a second plate, which contains immobilized substrate protein synaptobrevin. And if active light chains are present, then the synaptobrevin will be cleaved and the resulting cleavage fragment can be detected in an antibody-mediated reaction. So this binnacle assay scans several specific properties of active toxin molecules. So in order to generate a signal in this assay, the molecules must first of all have an intact binding domain in the heavy chain. Then they must have a functional protease domain in the light chain. And in addition, both chains must be separable by reduction. So when using this assay, so we found that the signals induced by non-spiked toxoid did not exceed the blank value, even when the toxoid was applied in high concentrations. And when the same toxoid was spiked with active toxin, we found a clear dose-response relationship that allowed sensitive toxin detection. So, no, that was wrong. Let's go back one. So compared to the single assays that I showed you before, this combined assay allows a much better discrimination between active toxin and inactivated toxoid. However, it should also be noted that not all toxoid are equally suitable for binnacle testings, as toxoids from some sources induce some elevated background signals. I'll come back to that later. Yeah. Now I'll show you some data for the tetanus binnacle assays that resulted from an international collaborative study. So this collaborative study, named BSP 136, was organized by the European Directorate for the Quality of Medicines and Healthcare in the context of their biological standardization program. And my colleague Beate Kremer and I contributed to this study as project leaders. And several manufacturers of vaccines for human and veterinary use, as well as national control labs, took part in the study. And each participant was supplied with a detailed assay protocol and with critical reagents. And as test samples for the study, we used tetanus toxoids that were spiked with toxin in order to mimic insufficiently inactivated toxoids. And each lab was asked to perform 3 to 4 binnacle tests, and the data were then analyzed by EDQM. And the aim of this study was to characterize the applicability of the method for the toxicity testing of tetanus toxoids. So in the first part of the study, 19 labs took part, and most of them were able to generate clear dose response curves for the toxin samples. However, we also found a high variability between the labs. So some of them were able to detect the toxin already in concentrations below 1 nanogram per milliliter, but some other labs had difficulties in detecting even high toxin concentration. And based on these results, we optimized several steps of the binnacle protocol and took care that only pre-qualified reagents were used for the assay. And the goal of the optimization was to achieve a better standardization and to remove potential sources of variability. And the resulting improved assay protocol was then tested in a second part of the collaborative study. So in this second part, 9 labs were involved. And in this part of the study, we only involved manufacturers from European countries, just in order to make the shipment of the reagents easier. And you can see that using the optimized protocol, all participants were able to obtain clear dose response curves. And all labs were also able to detect low toxin concentrations. So during analysis, the detection limit was analyzed by a cutoff-based methods. So all signals that were above the mean blank value plus the 3.3-fold standard deviation were considered as toxin positive. And this analysis showed that all laboratories were able to detect toxin concentrations of 0.3 nanogram per milliliter, and most of the labs were even able to detect 0.1 nanogram per milliliter of the toxin. And unfortunately, there's not much reliable information available about the sensitivity of the guinea pig test for tetanus toxin. But based on literature reports and in-house experience, we estimate that the detection limit of the animal test is probably between 0.1 and 1 nanogram per milliliter. So the detection limit of the tetanus binnacle assay and of the animal test are probably in a similar range. And this is an important prerequisite for the use of the binnacle assay as alternative to the guinea pig test. So, okay. When we then analyze the variability of the data obtained in this second part of the study, we found that with the optimized protocol, reproducibility, which includes the intra-laboratory as well as the inter-laboratory variability, was 13%. And this level of variability is in the range that is commonly reported for immunochemical assays. And so assay optimization had really helped to reduce the variability to an acceptable range. So now I'll come to the conclusion and the outlook. So the collaborative study had shown that the binnacle assay allows a reliable detection of active toxin. So we found that the method is well transferable to other labs, as all participants were able to successfully perform the method. We found that the detection limit is equivalent to the estimated detection limit of the in vivo test. And the variability that we found is in an acceptable range for a multi-step assay. So the assay really fulfills fundamental prerequisites for a suitable in vitro method for toxin detection. It should be noted, however, that the binnacle assay may not be applicable to all toxoids, as toxoids from some sources induce high background signals. And we think that the different behavior of toxoids in the assay is probably related to differences between the toxin inactivation protocols that are used by different manufacturers. But we, in our experience, toxic batches from the same source usually show a low batch-to-batch variability in the binnacle assay. So, but however, we are convinced that for suitable tetanus toxoids, the binnacle assay may really represent a good alternative to the safety test in guinea pigs. And now as an outlook, so based on the results of the collaborative study, discussions about the inclusion of the method into the European Pharmacopoeia as alternative to the guinea pig test for the absence of tetanus toxin are currently underway. And a crucial point for the usage of the method is the careful validation. So before the binnacle assay can be used for the quality control of vaccines, the users really need to validate the assay for their specific purpose and their specific toxoid product. And a key point for the validation is that it must be really carefully checked that non-compliant batches or bulks are reliably detected. So it's really important to make sure that the switch from in vivo to in vitro testing has no negative impact, but rather provides a solid basis for ensuring high product quality and safety. And if you are interested in learning more about the method, a webinar about the tetanus binnacle will be held in November. And this webinar is organized by EDQM, and more information about the event can be found on the EDQM homepage. So finally, I would like to thank all the members of our project team at the Paul-Ehrlich Institute, and also all other people and organizations that supported and the binnacle project and contributed to our results. And I want to express my special thanks to the colleagues from EDQM for the great support related to the tetanus collaborative study. And thanks to all of you for your attention.
Thank you, Maike, for your presentation. I think that there are a couple of questions in the Q&A. There is a comment about the application of Pinnacle maybe to other microorganisms. So I know that you use it for the botulinum, right? Mm-hmm. If you can comment about the extension of the applicability of the Pinnacle to other type of products in addition to tetanus.
Okay, so we already adapted the method also to botulinum toxins, at least botulinum toxin serotypes A and B, which are the ones which are relevant for pharmaceutical purposes. So yeah, that has been done by us. So the whole method, it might also be able to, adapt it to other botulinum serotypes if needed, or if that's, but it was specifically adapted to toxins which have a binding, of course, and an endopeptidase function. So I'm not sure, it would have to be checked if it can also be applied to other kinds of toxins which have a different mode of action. We have not tested that so far.
Thank you, Heike. And then there is another comment about the fact that the assay is going to be giving a good picture about the effectiveness of the vaccine, but it might not probably be reliant for, I think, release. Do you have any comment on this?
Effectiveness of the vaccine. So our assay is mainly was designed as a safety test, not as a potency or effectiveness test. Just it was developed as an alternative to this test for the absence of residual toxin. And we think that at least for these suitable toxoid products, it will really be a good alternative, and also, yes, suitable for batch testing purposes, but it was not, its concept was not as an effectiveness or potency assay.
Yeah, it's for the safety. Thank you.
Yeah.
Thank you, Heike.
You're welcome.
Okay, so in the please, Natalia, we need to move to the next presentation. So can you type your question into the chat and or in the Q&A, and we will try to address it. So thank you very much, Heike. We are happy to move to an interesting case studies about the possibility to use cell-based assays for the tetanus toxin. And we have online Andrew Peden from the University of Sheffield and the Shalini from MHRA that are going to present the work that they have been doing together on this specific assay. So, Andrew, thank you. We can see your presentation. So the floor is yours.
Can you hear me?
Yes, I can hear you as well. Yes, thank you.
Okay, so thank you very much for the invitation today. So I guess it's going to be slightly different from some of the presentations that you've seen so far, but I should thank Heike for giving a very good introduction to the biology of tetanus toxins. So I'm a professor of translational cell biology at the University of Sheffield, and my group is interested in the basic biology of snare-cleaving clostridial toxins. And so today's talk, we're going to tell you about a little bit of the work that we've been doing in collaboration with MHRA to develop novel cell-based assays for measuring the biological activity of tetanus toxin. So I'm not sure if Shalini wants to introduce herself now or to do it later. What do you want to do, Shalini?
I'm happy to go now, if that's okay.
Okay.
Yeah, so I'm Shalini from the MHRA, and I work with Paul Stickings, and we are long-time collaborators with the University of Sheffield and Andrew and Kira's lab on, looking at development of safety testing for tetanus toxoids.
And I should say, I'm not sure if Kira's going to say hello or not. Kira is also a postdoctoral researcher in my group who's done a lot of the work which I'm going to present today. But also, I should say that Myself and Kira have just recently formed a spin-out company called Sansanima from the University of Sheffield with the aim to try and help them in implementation of these cell-based assays for the detection of these types of toxins. So Heike, as I said, gave a really nice introduction to this area. So obviously, botulinum and tetanus toxins are some of the most potent toxins known in in nature. And so very small amounts, nanogram amounts, are basically lethal. And so there's very high levels of testing required to show that these molecules are safe for the various applications which they're used in. And so these tests are not only performed by the manufacturers making these molecules, but also government regulatory bodies. And so these can range from obviously potency testing, so measuring how much of the active toxin you have. So that would be in the space of botulinum-based therapeutics. Residual toxicity testing, which is obviously Heike's very nicely introduced with the tetanus toxoid experiments, antitoxin testing, or the production of antitoxin testing, and then I guess neutralization or potency assays to measure how well the vaccines are actually working. And the sort of gold standard for these assays is obviously either mouse or guinea pig-based bioassays. And these have some of the highest severity limits which are currently allowed for animal testing. And as people have touched upon, they have very high levels of variability. They're expensive, and due to the variability, there's very large numbers of animals used. And certainly in the botulinum space, when you combine that into the tetanus as well, you know, hundreds of thousands of animals are used. So there's a really strong ethical imperative to move away from using these assays, but also there's quite a strong imperative due to the financial cost to the manufacturers using these assays as well. And so the question you might be asking is, so why is somebody from the University of Sheffield interested or trying to work on this? So we were approached originally by Thea Sisaderic and Paul Stickings from the MHRA, and actually a former colleague, Bazbek Davletov, who's now retired from the University of Sheffield. And we were asked, is there a way that we can take our knowledge of the basic biology of these toxins and try and use them to sort of develop novel approaches or cell-based assays to replace animal testing? So, and obviously in the areas of potency, antitoxin testing, neutralization, and residual toxicity testing. So I'm just going to talk a little bit about the biology, and some of this hopefully is not going to completely overlap with what Heike said. So botulinum and tetanus toxins, they're taken up into neurons, motor neurons, and actually the botulinum toxins. So in this process, the toxin binds to the surface of the neuron, is then endocytosed, it then translocates, and then there's a cleavage event. So there's basically 4 key steps which occur before you get intoxication. And for tetanus toxin, there's a retrotranslocation event, which involves the inhibition of inhibitory or intoxication of inhibitory neurons. And this leads to basically a form of rigid paralysis, or actually if you're a botulinum toxin, you end up with infected with the botulinum toxin, you end up with a flaccid paralysis due to these slightly different mechanisms of action or the different neurons which they intoxicate. And as Heike mentioned, the botulinum and tetanus toxin, they cleave a molecule called VAMP2 or synaptobrevin, depending on which labs you're originally from, whether you're from Europe or from America. So VAMP2 gets cleaved, and this then releases a fragment of the protein, which means the VAMP molecule is unable to interact with other snares and drive synaptic vesicle fusion. And so one of the really big challenges when trying to develop cell-based assays for studying the biology of tetanus toxin and botulinum B intoxication is the VAMP molecule, when it's cleaved, becomes degraded. And so there actually isn't any of the molecule left after it's been cleaved to allow you to assay. And so that's been a really big challenge for people trying to develop cell-based assays in this space. And so what we've done is we've used our sort of understanding of the basic biology of these molecules to develop a simple and robust assay for measuring the activity of botulinum and tetanus toxins. And so in this assay, there are 3 basic components. So if you look to the left, we use neuroblastoma-derived cell lines. And so these cells are able to efficiently take up the toxins, botulinum and tetanus toxin, when they're differentiated. We've then engineered the VAMP molecule, the synaptobrevin molecule, to have a nanoluciferase tag. And so that basically has 2 roles within the assay. So Firstly, it allows us to detect the presence of the molecule, the VAMP molecule, when it's cleaved. But more importantly, what we discovered is that when we add the tag to the molecule, it basically protects the construct from degradation, or the VAMP molecule. So it solves this long-standing problem when trying to measure the activity of these toxins, because the VAMP normally would disappear, the synaptobrevin is degraded or broken down within cells, and so this protects it. And then finally, we use cleavage-specific antibodies, a bit like HEICA, to capture the fragment which has been generated. And in the initial assays, we actually used an antibody which had been generated by the MHRA. So when we combine these things, all these components together, we have a very simple assay for measuring the activity of these toxins. So in a standard assay, we would differentiate the cells for 3 days to make them sensitive to the toxin. We then incubate them with the toxin. We then break the cells open, and then we basically perform a one-step ELISA assay to measure a luminescence reaction to measure the activity of the toxin. And so in the first instance, when we did this, the cells that we generated were actually only sensitive to botulinum B. And this is an example of some data which was generated between Kira and Shalini. So we transferred the cell-based assay to the MHRA, and they performed an antitoxin experiment. So if you look to the left, we have the controls, so the non-treated control. We then treated the cells with botulinum B, and then we incubated them with an antitoxin. And hopefully that you can see that we see a very good inhibition or loss of the signal, showing that the cell-based assay is faithfully recapitulating the biology of the toxin. So we then work from Kira, basically, was able to identify cells, or neuroblastoma cells, which were sensitive to tetanus toxin. And this is really the sort of first iteration of the assay. And so we transferred these cells again to the MHRA, and this was published as part of a VAC2VAC study. And so in this experiment, what we did is we incubated the cells with tetanus toxin. And so you can see in the sort of blue bar, we get a nice response. And then we incubated it with various different anti-tetanus antibodies to look to see if any of them had any neutralization activity or could inhibit the biology of the toxin. And so you can see there's, from different of these antibodies, have different effects. So again, suggesting that, the technology that we're developing was potentially useful for measuring the biological activity of the toxin. So in the final iteration of this assay, so one of the things that we realized that actually we needed to generate the critical reagents of the assay, we needed to generate basically sustainable versions. So for a lot of the cleavage-specific antibodies, which are used in the capture step, they're generated as rabbit polyclonals. And we realized that that's actually not going to be particularly useful if we want the assay to be adopted more widely, and for it to be sustainable. So one of the things which I've been involved in is generating recombinant capture antibodies to the cleaved SNAP, cleaved VAMP2 or synaptobrevin. And so what we've done is we've immunized animals with basically peptides which mimic the cleavage of these molecules, and then we've pulled out antibodies in which we've cloned these antibodies subsequently. And so this is just a little example of some sort of validation data. And so what we've done is we've taken our neuroblastoma-derived reporter cell line, we've incubated with botulinum toxin, and you can see that only in the samples treated with the toxin do we see very nice receive the cleaved VAMP2. So sort of indicating that these antibodies are going to be useful for working in these recombinant capture antibodies. And so in the literature, it's generally thought that the limit of detection in guinea pigs is about 0.67 picomolar or 0.1 nanograms per mil. And you can see in the cell-based assay, we're certainly getting research grade cell-based assay. We have very good levels of sensitivity, which are probably below that of a guinea pig. So just to summarize this little bit of the talk, which I've been telling you about, so hopefully what I've shown to you is we have a simple quantitative luminescence-based assay for measuring the activity of botulinum and tetanus toxins. And most importantly, it covers all the biological steps of intoxication, so binding, endocytosis, translocation, and cleavage. This assay is obviously slightly longer than an ELISA, so it takes about 7 days to perform, which is obviously far shorter than the animal-based models. And then we generated recombinant capture antibodies, which actually will mean that this assay is fully scalable. And so these antibodies, we're hoping that we will make these available to the MHRA. So I'm now going to hand over to Shalini, who's going to tell you about the study that we're doing, which is being funded by the NC3Rs, to basically see if we can, this assay is useful testing real-world samples, or samples generated by manufacturers.
Thanks, Andrew. And thank you, Laura, for inviting us to speak here. So we'd like to tell you a bit more about the tetanus cell-based assay collaborative study that we've set up. So this is split into 2 different phases. So I'll start with phase 1. So we've approached tetanus vaccine manufacturers, both human and veterinary, to see if they will donate tetanus toxoids for us to test in the cell-based assay. So to do this, we're doing parallel testing at the University of Sheffield and at MHRA as well. And so we're going to be using these bulk tetanus toxoids that have already passed the in vivo requirements for the testing in terms of the absence of tetanus toxin and irreversibility of the toxoid as well. So in phase 1, the focus is going to be looking at the suitability of the assay for testing tetanus toxoids, looking at detection of the toxin spiked into toxoid, and also looking at the limit of detection of the assay. And the plan from phase 1 is to publish the anonymized data from manufacturers' tetanus toxoids. And then we will also be looking at specificity of the assay using tetanus antitoxins as well. So this phase 1 has already commenced. And then I'll tell you now a little bit about phase 2, which is what we've got planned coming up. So phase 2, this would be technical transfer of the tetanus cell-based assay using the nanoluciferase VAMP2 transfected cells under MTA conditions to vaccine manufacturers. And with this, there would be training and critical reagent provision, including the monoclonal antibodies, for conducting the full cell-based assay, including the luciferase assay. So the data from this part of the study will be generated by the tetanus vaccine manufacturers, and it will give us a real-world view of how applicable this assay can be to the tetanus vaccine community. So next slide, please. So this slide is just a brief overview of our participants so far in phase 1. So we have a number of human tetanus vaccine manufacturers who are on board, and also we've got 2 veterinary tetanus vaccine manufacturers as well. And we just asked, as part of our initial setup for phase 1, just for rough estimates of the number of animals used per year for tetanus toxoid safety testing. And you can see it ranges from the hundreds to the thousands. And this is only a really small section of the safety testing that occurs for tetanus toxin. And it's worth just noting here that the tetanus toxoids that we've received so far, they really range in concentration in their LF per mil. And so as part of our SOP developments, we'll be factoring that into the cell-based assay study. Yeah. Okay, so next slide, please. Okay, so at the MHRA, we're really interested in standardization of this assay and how we can add value to the tetanus cell-based assay at the University of Sheffield. And so one of our ideas that we are now taking forward is to generate a tetanus toxin reference reagent. So we've previously experienced some variability with commercially available tetanus toxins. And so we'd really like to provide a standardized tetanus toxin that could be used for as a positive control for safety testing of toxoids. And so this toxin reference reagent, it could have utility in the tetanus cell-based assay, but also perhaps in the barnacle assay as well. So the tetanus toxin material that we've received has been donated by a tetanus vaccine manufacturer, and it has come with accompanying in vivo data. So we have LD50 data and PD50 data on this tetanus toxin. And the plan is that we would make this new reference reagent available for all to purchase through the MHRA and IBSE catalogue. And as you'll have heard from our talks and from previous speakers, there are some big challenges to do with making this reference reagent, not least that tetanus toxin is highly toxic. And so that has consequences on the handling, the formulation, the freeze drying or preparation of the toxin, and even international shipping. So these are things we are considering as we set up this reference reagent. We're really interested in understanding better the biology of tetanus toxin, particularly its stability, and monitoring that stability with this reference reagent. So thank you, and I'll pass back to Andrew for the rest. Thanks.
Yes, I'd just like to thank you, obviously, the people who've been involved. So obviously, Basbeck and Thea started this project, and obviously with Paul Stickings, and obviously the team at Sheffield who have been involved in this. So Kira, who's actually has really driven a lot of this and has actually been involved in the sort of formation of our spin-out company. And really, a lot of this, we wouldn't have been involved in this really without the MHRA suggesting, we might be, our expertise might be helpful to try and solve some of these problems. And then finally, most of this work has been supported by numerous grants by the NC3Rs over the basically the past 10 years. So yeah, so thank you very much. Thank you for your attention.
Thank you, Peter. Thank you, Shalini. Also, thank you, Tara, for the work done and for the presentation. There is a question on the Q&A asking about the possibility to use vero cells or other type of tissues for the toxicity. So we have been seeing your experience with.
Yeah, no, unfortunately, well, not for the botulinum and tetanus toxins, because they exquisitely target neurons. So that's one of the massive challenges in this field, is to actually get. So a lot of people do work with sort of primary neurons, obviously isolated from animals, and then you can also do work from stem cells. But the time which takes for the stem cells to generate sensitive cells, it can be weeks. And so these neuroblastomas sort of overcome that problem. So we're able to generate sensitive cells within a few weeks, and we're not having to rederive them all the time. So yeah, no, you can only use neuronal based cells.
Thank you very much, Andrew, for answering the question. So please add any other question in the Q&A for Andrew, Shalini, and Tara. They will be happy to answer in written. And I'm happy to move to another topic, but definitely very important and very, very, very recent as well, because we are moving to talk about the next generation sequencing technology as a way to test for the adventitious viruses. And we have here Blandine de Sanvis from Boehringer that is going to talk about the recent changes in the European Pharmacopoeia on the application of this technology. Blandine, the floor is yours.
Thank you. So I will share my screen.
Please, thank you.
So, is it okay like that? No, it's not that.
Perfect. We can see it. Thank you.
Yeah. Okay. So thank you for giving me the opportunity to share the work we have done for next generation sequencing. I will talk about advantageous viruses, and I will try to give you an overview of the key resources for regulatory approach for NGS or HTS. So I'm part of Boehringer Ingelheim Animal Health, and I'm working in analytics for several years, and I'm a member of the EDQM HTS working party. So next slide. So I will give you a brief introduction of an NGS, HTS. I will give you an overview of the extraneous agent testing for vaccine or viral vectors. And I will give you an overview of the work we have done to write the new chapter on NGS at the EDQM. So first, a brief introduction on NGS. So NGS is for next generation sequencing. It's also called HTS for high throughput sequencing, or it is also called MPS for massively parallel sequencing, but it all, the three words says the same. Thanks. So what is NGS? NGS is, I told you, next generation sequencing. It's a sequencing technology capable of sequencing all the nucleic acids extracted from a biological samples. So it gives you millions of reads. So it's been sequenced from a library of nucleic sequences, whether it comes for DNA or RNA of a mixture. It's an extremely fast method. It's a powerful tool for identifying known and unknown viruses. And as you imagine, there is a large number of applications. There is also several technologies. So you heard probably about Illumina, iTorrent, Nanopore, PacBio. We talk about short read or long read. And this technology is able to detect any potential viral contaminants in a biological sample. So this is an overview of the NGS workflow for the preparation. So you need to put under control several steps. So you have the preparation of the sample, you extract DNA and RNA, you have a step of enrichment of library preparation. Then you have the sequencing itself. So at that stage, you need an equipment. It could be a Illumina or another method. And then you have a huge part of bioinformatic analysis. And in that time, you need you have a huge work for analysis the reads. For master seed testing, usually we perform a random sequencing of all nucleic acids, DNA or RNA. So I will give you an overview of the current advantageous testing. So this is a workflow for the vaccine development. And as you know, we need to control the starting materials for the absence of contaminant. It could be master cell bank, working cell banks, master virus seeds, raw materials. And we need to demonstrate the absence of contaminant. It means bacteria, fungi, Mycoplasma assay, and of course, virus assays. And these assays are submitted to regulatory agencies for vaccine registration. And in this figure, you have an overview of the complexity of the classical seeds for purity testing. So we have to deal with cell banks or viral seeds, and we use permissive cell lines, detect their cells, and we use non-specific testing and specific testing to have the largest as possible analysis. The limitation of this method is that it takes time. It's a long analysis. We know that some cells are not permissive to virus, so there is a limitation of the method. And to perform this testing, we need neutralizing monoclonal antibodies. We could be an issue in some circumstances. So recently, the approach for testing changed. There is a new European Pharmacopoeia chapter that has been revised. The chapter 525 has been revised and applied since July 2020. And the philosophy now is to first start to do a viral risk assessment to define and to justify for not conducting the search of extragenous agents. And for that topic, we need to have a good knowledge of the production process. We need information of the raw material use, the production process, if we use, for example, dilution, treatment, and so on. And we need certificate tested animal-derived material. And when we have do this risk assessment, you perform the testing itself, and you define the agents that you need to test. This is a classical method, but more and more there is the emergence of NGS as it's a powerful tool. And this method is extremely powerful method for the characterization of the seeds. And as I told you, it allows to identify known and unknown advantageous virus without needing a prior knowledge of the contaminant agents, whatever the virus. And the interest of this method has been shown for two recent in biologicals in two recent work. It has been discovered the PCU1 in a licensed rotavirus vaccine, and another rhabdovirus has been also identified in SF plus cell line, which is used for baculovirus express system. So now I will move to the new regulation for this NGS, which is I will give you more details on this text. So it has been shown. Sorry. So as I told you before, we need to define the testing strategy for adventitious agent, but there was no description on HTS and NGS, no guidance for the validation of the method. That's why EDQM HTS working party has been put in place. It has been possible because more and more it has been developed regulatory standards, appropriate reference standard developed by FDA and adopted with WHO. And there is also the development of new database, reference viral database. And as for your knowledge, the draft has been put in public consultation until end of this year, the 3rd of June. What we'll find in this new European chapter 2641, so we'll find a huge description of the HTS and NGS methodology for the detection of viral exogenous agents in biologic products. So the scope is large. It concerns vaccines, recombinant proteins, viral vectors used for gene therapy, cell-based preparation for cell therapies. So to the right, you can see in the table, you have a different description of the steps used for the NGS, and you have a huge part which concerns the method validation, and small part on targeted HTS. So this is an overview what we'll find in this chapter. In the description of the method, you have a huge figure explaining how to define the strategy. So first of all, you need to define the HTS strategy for viral agents, and it depends on the choice of sample types on the production stage. And as shown in the figure, you have different approach for sample preparation. It could be genomics, it could be viromics or transcriptomics when you target only viral RNA. And in this schematic representation, you have description of all the stage, sample preparation, nucleic acid extraction, and so on. So in the next part of the chapter, you will find information about sequencing and bioinformatics. So of course, you need to select the most appropriate sequencing technology platform. You should take account the sample types, the sequencing depth and coverage, accuracy, and wavelengths. And you have also an example of the bioinformatics workflows that you can use. So of course, it's very important to have a good quality of raw reads. You can perform either one sample per run, or you can also use multiplex sequencing. There is a description on the strategies that you can put in place to reduce the number of, to facilitate the bioinformatic analysis. There is also a topic on a database. So you have the choice of the database, but you need to have a well-characterized database. And of course, also you can use different pipelines. It could be custom-made or commercial pipeline. So the workflow for NGS is complex. So there is description of each steps. Of course, there is a part concerning the evaluation of the results and the follow-up investigation. So the validity of the HTS should be based on recovery of controls, one or more internal control, or the results of a control sample run in parallel. And when you obtain a positive for the interpretation of your result, you need to define your criteria for distinguish a true positive against a false positive viral signal. You should put in place appropriate control to avoid cross-contamination for the epsilon. And when you have a true positive hit, you just target genome and nucleic acid. So you need to demonstrate that with a laboratory-based follow-up that you if you find a sequence, it's associated with an infectious virus. And in the text, you will find a guidelines for the validation. So of course, you need to demonstrate that the method is suitable for the intended purpose. It could base upon the sample type and testing approach. So it depends on the types of sample. It could be cell substrate, cell bank, virus. HTS is a qualitative limit test. So the parameters to assess is specificity, including the breadth of virus detection, and limit of detection. And as I explained at the beginning, the workflow for the NGS is very complex. There is several modules. So you need, of course, to perform an end-to-end assay validation, but you could also subdivide the workflow into a module, especially when you will have to revalidate or to change some element in your workflow, in your full workflow. As I explained also, it's very important to have a very well characterized spiking material for validation. For genomics or viromic approaches, the reference standard should be viruses representing viral diversity in terms of structure, nucleic acid type, DNA, RNA, single strand, double strand, etc. Genome size, morphology, they should be well characterized. And it's recommended to use the panel developed by FDA and adopted by the WHO International Reference Standards. And as conclusion and next step, so I can confirm that the more and more companies use NGS or plan to use NGS for purity testing. There were the recent years huge boost with the mRNA vaccines and new therapeutics. Most of the essay concern Illumina platform, but it's open for other platforms also. The HTS, as I explained, is a part of the testing strategy for the current chapter 525 and for advantageous agents. And it could substitute or replace multiple in vivo or in vitro tests. So it contributes to to reduce the use of animal or animal-derived assays. And this future general chapter 2641 on NGS will provide a detailed description of the technology together with the validation guidelines to support others to implement these new technologies. And if you would like to know more about the NGS, there will be a first conference on NGS organized by EABS in Frankfurt in December. And there is also a one-day training on the method. So I would like to thank the people from EDQM, the HTS Working Party, and Johannes Blumel from PEI, and several people from EDQM leading this project, and Arifa Khan in US FTA. Thank you for your attention. I will be happy to answer to a question.
Thank you, Blandine. Yes, indeed, we have more than a couple of questions, but I try to summarize them. So we have a question about if it's possible to use NGS if you don't have any metadata.
If you don't have what?
Metadata.
You need to analyze you will have metadata. You will have a lot of data that you need to analyze. So there is different strategy, and you can reduce the number of reads that you obtain, but usually you have a huge number of data. So you have the bioinformatic part is a huge part. In any case, you can perform, for example, pre-treatment, or you can work on the way as you prepare your sample, but at the end you will have a lot of data for sure.
Mm-hmm. Thank you very much, Blandine. Then we have some questions about the regulatory acceptance. So what is actually your view on the complete transition from in vivo data requirements for adult tissues agents to NGS as a platform to completely replace animal use for this type of testing? Because there is still a lot of expectations, I suspect, in the low and middle income countries to probably perform both.
So in fact, I think the NGS method should be, for the moment, complementary to the existing testing. and it allowed to suppress a lot of testing. The difficulty with this method is to fully validate the method. It should be the case in human for some company validate fully the method, and in some cases it has been used as unique method for testing, property testing. But it could be, we can imagine also to use a combination of NGS and other testing. So, and I know that it has been accepted by regulatory authorities.
Thank you very much, Blandine. One last point about the availability of positive controls for the viruses in the NGS for the assay validity. So there are guidelines in order to support the validation of the NGS results.
Yes, exactly. Several viruses has been developed by FDA, and they will be they put in place stocks, huge stocks, and these stocks will be available for all manufacturers to validate the method. And the interest that we imagine that in the future all companies or manufacturers will use the same panel of reference stocks, and we can compare a result from one product to another product.
Thank you very much, Blandine. We answered all the questions. So thank you very much for your presentation, and looking forward to see the NGS technology becoming available to all and accepted by regulatory authorities from both the human and veterinary side. Thank you, Blandine, for being with us. And last but not least, we are getting to the end of our webinar. Thank you for still being with us this afternoon or evening or late morning. I would like actually to invite Elizabeth Bax from the Paul Ehrlich Institute to give some final remarks in order to complete this webinar and to have some take-home messages for all of us. Thank you, Elizabeth. We are seeing your screen, but in the presenter mode.
Okay.
If you can change it from the presenter to presenting.
Better now?
No, still like before.
Okay, then Laura, maybe you could present.
I can share.
Yeah.
Of course. Give me a second, and I'm bringing up. If you can stop sharing. Perfect. Tell me when you see your slides.
Yeah. Okay.
Thank you, Elizabeth.
Okay, thank you, Laura. Next slide, please, then. Let me briefly recapitulate what has been addressed this afternoon. There have been examples for international outreach. Laura reported on the VICH guideline initiative, technical recommendations for the transition to in vitro methods for batch potency tests. And Dean exemplified the impact of VAMUR chapter 5 to 14, even going so far that a 5 to 14 like WHO technical report series is to be expected. And Carmen gave an overview of the VAC2VAC project, which was a very successful cooperative effort of human and veterinary medicine. The achievements in terms of publications are still available on the EVI website in the internet. The address is given here. Next slide, please. Then we had 2 case studies, first on rabies, second on tetanus. Catherine, Benjamin, Peter, and Ignacio gave an overview of their efforts on establishing antigen quantification for veterinary rabies vaccines. Here we are in the comfortable situation to have the relevant epitope well established, and the established assays detect the immunological relevant antigen in the proper conformational form. But nonetheless, a single assay method turned out to be not suitable for all products. So we are now in the situation to have 3 different glycoprotein ELISA methods for potency testing, globally accepted and developed by different companies, starting with Merial, now Boehringer Ingelheim, MSD, and Zoetis. Catherine pointed out that it is very supportive to have historical data available and to use the consistency approach to establish release and end of shelf life specifications, and also to establish a reference without additional challenge testing. And moreover, link with efficacy and safety study very much facilitates the procedure. So then the case studies on tetanus. We had 2 examples for, or 2 presentations on ELISAs for antigen recovery. ELISAs are applicable to a broad range of products, but here again, product-specific measures may be needed, especially with regard to the desorption step. Then we have 2 examples of toxicity testing, starting with the Binnacle, which is the most developed assay, I would say. It detects active tetanus toxin based on their receptor binding and proteolytic characteristics, and may thus be a suitable alternative to replace absence of tetanus toxin testing in guinea pigs. A BSP study, or 2 BSP studies actually have been finalized, and now discussion is ongoing about inclusion of the method into the European Pharmacopoeia. Heike pointed out that product-specific validation is needed, since method may not be applicable to all toxoids. And then Andrew and Shalini introduced us to their cell-based luminescence assay, which they explained is slightly more complex. Slightly more complex and reflects the full intoxication model, and is based on the use of recombinant capture antibodies. Here, a first collaborative study is just ongoing, and future will tell the exact benefits of these 2 different functional and maybe partly complementary methods we have now available. Next slide, please. So, last presentation was from Blondine, who walked us, or who introduced us to next generation sequencing, which could be a possible at least complementary testing strategy for extraneous agents testing, according to European Pharmacopoeia chapter 525. She walked us through the new general chapter 2641 to come next year. and pointed out that this new monograph will provide a detailed description of the technology and will also give guidance on validation requirements. She pointed out that there's a real need for regulatory standards and appropriate databases, microorganism databases, and that positive hits require additional methods and a detailed laboratory-based follow-up. Next slide, please. So what could be regarded as key aspects taken from the presentations we have had this afternoon on the laboratory side related to methodology, it was pointed out that methods should be science-based and data-driven, that it is necessary to know and define critical quality attributes. Here, I really would like to draw your attention to a publication coming out of VAC2VAC, given a very nice overview about the use of the and application of the consistency approach. Moreover, the suitability of methods through all steps of vaccine production and vaccine life cycle is key, and also the applicability of methods in various scenarios depending on the matrix, like multi-component or adjuvanted vaccines. Stability indicating potential of the methods is supportive, and methods should be robust and transferable to GMP settings. And it was pointed out several times that product-specific validation is necessary in many scenarios, and the benefits are to evaluate new technologies were emphasized. With regard to reagents and reference material, their availability, sustainability, and their characterization was emphasized to fulfill or to reach our common goal, namely international, yeah, that would be the more humble wording, international or even global cooperation and alignment and regulatory acceptance, starting with sharing of information and publication of results. Last not least, it is essential to keep laws, regulations, and pharmacopoeia up to date with regard to state of science, and to develop new testing concepts by innovative thinking. So these were my closing remarks, and I would also like to thank you, Laura, for setting up this kind of webinars, because I think they are really a good starting point to get this exchange and to bring people together. So thanks a lot.
Thank you very much, Elizabeth, for your closing remarks to highlight it, how important it is continue the collaboration, and that's what we really hope to continue doing through these webinars. So I really want to thank you all, speakers and participants online, for being with us, and please do monitor our AFSA collaboration channel on LinkedIn, and we will make sure to communicate to you about upcoming events and future collaboration opportunities. So thank you very much to you all. Presentation and recordings will be available in a week time on the AFSA website. So thank you, and I wish you a nice rest of your day. Bye.